So, good morning to all of you. Thank you for joining in time. I already saw that some people started to answer the WooClap. So thank you for doing this. Those who didn't, please scan this QR code on your screen and please answer the question, what is the biggest challenge facing healthcare in the coming decade? I saw that somebody had some other health challenge in mind. If you did have another big or major challenge in mind, don't hesitate to put your answer in the Q&A that you will find here in the Zoom, and then we can track that and I will comment on this at the end of the day before we all say goodbye. Before I introduce what we will handle today in Module 2, I will go over some practicalities that are important for you to know. So you are here in a virtual environment. So it is very important, as I told you last week, 2 weeks ago when we saw each other in person, that this is as much as interactive as possible, because this is nice for us as a team, I3H team, but also and particularly for the speakers who are here presenting for you. If you have questions, don't hesitate. If something is not clear, an abbreviation, or you have a question that really could stimulate the debate, then please put it in the Q&A, and you can be promoted to become a panelist, and then you put on your camera and we can have this discussion with the speaker live. So please be as interactive as possible. Here again is the QR code for the question. It will come back at the end. So today we are in the Module 2, the online module, the first time we meet online. I hope that some of you already had the chance to look at the e-learning module that is part of this second module by Matthias Neet, and it's really about the basics of healthcare economics. If you already have a background, then you can definitely skip it. Voila, this is this. And then half of you already answered whether or not you would like to participate in the certificate track. I will, if you didn't do so, next Tuesday by end of business is the last time that you can choose. If you didn't choose by then, we will consider that you only do the 5 ECTS, because I explained this in the first module. All of you will do the online MCQ exam, but for those who choose to do the certificate track, you have the option to really do a group work. You are expected to participate in the on-site workshops, and you will work in group and defend before an interdisciplinary jury. And of course, you can imagine that we need to know, one, how to compose the most interdisciplinary groups for doing this, and also who to assign, because we have jury members who are more interested and knowledgeable in particular topics, of course. To make sure that we have a schedule that fits their schedule, we need to know upfront your topic, if you participate, and when you will participate. Voila. The MCQ exam, and this is, we will go over very quickly for all of you, so it's recorded and you have it. It will start in the Université Virtuelle, so the online platform where you find all the slides, where you find the recordings from the moment we get them. So the slides from today will, most of them will be online by tonight. The recordings, it takes time, of course, as you can imagine, to process them. At the latest, it will be online Wednesday. So we do our utmost to be as quick as possible, but yeah, voilà, more we cannot do. The online exam is at 10 a.m. sharp, so you have 2.5 hours to participate on the 8th of May. And you will get an insight in the type of exam questions, as I told you. After the module, after this whole program, you get a small evaluation. And if you give us some feedback on the program so we can continue to improve, then you get an insight in some sample questions, and you will understand how we assess what you grasp from this program. So the case studies are really aimed to creating ingenious, unorthodox solutions for some of those health challenges that we already discussed. And the idea is really, as I explained last week or 2 weeks ago, that you work in interdisciplinary groups, that we try to balance the level of so master students together with PhD, postdocs, and potentially even professionals. You give a top 3, and we make the best options to create balanced groups, interdisciplinary groups. And these topics, as I explained, are very broadly defined. If you don't find a focus, a specific geographic area that you want to work on, or a specific focus, like a specific therapy or a specific disease area, don't hesitate to come back to me, and maybe I can help you. In any case, I am there for you. Sometimes I will say I will send you some background material to start from, but it might be that just by I would say searching on the internet, and particularly PubMed, of course, that you will find an angle. Again, here, the QR code to choose whether you want to do this extra certificate, which is, of course, a validated certificate that you can add on your CV to show that you are knowledgeable afterwards in healthcare innovation. These are the dates that you will then follow. In any case, for all of you, the online exam is on the 8th of May. And then, so it's really about defining the limits of this broad topic that you will be assigned to, and that you, as I told you last week, think from your background, whether it's biomedical sciences, medicine, economics, maybe engineering, that you think, what can I bring to add value to the group? You can focus, for example, on 1 or 2 interventions that you will work on out in detail, because this whole group work, and you will be 3, 4, or 5 people, depending on the number of people that choose a certificate track. You work in detail, but the detail is in 10 pages, so excluding references and appendices. But in 10 pages, you need to really come to define your topic, and to show that you understood what the challenge is and how you can bring a potential solution that we then can discuss. It's scientific work, of course, so the idea is that your references are really, really accurate, that you try to work with the most up-to-date references, that you first specified your topic, your background, and that you then in the background focus to defining your research question, that you work with a specific methodology. And if, of course, you don't do lab work and you don't have to define what ingredients you use for your experiments, but you will have to say where you found your information. I will also put online a form wherein you declare whether or not you used artificial intelligence. So most of you will use ChatGPT or Answer This. I don't know what program you will use, but you need to declare and to say how you prompted the system, so we can follow what you did in the group. Every year, we get complaints of people saying, I did the whole group work on my own, or there was a free rider. Please, this is really, if you decide to participate in a group work, you will all defend. So any member of your group will have to present. If you don't present, we will challenge you. We will ask you the questions to really find out whether you collaborated with your group or not. Please also make it a group work, not assigning tasks where nobody discusses with each other, because it's really clear for us too if you only focused on one particular aspect of your case study, and this doesn't really reflect the group work. So learn to collaborate. Voilà, this was more or less all with respect to the practicalities. And so this is our program for today. Now, by looking a little to what we've seen last week, we will have a look on what we can expect for today. And we have a busy program. During the first module, you already got a flavor of what this course entailed, and we highlighted quite exhaustively different health challenges and their relation to socio-economic, political, and environmental challenges, where access and affordability is at the center of many of the global healthcare challenges. We discussed the cost of innovation, and innovation is very expensive in healthcare. Research and development costs are high, manufacturing can be complex, and with the IP defining who can produce and who cannot, reimbursement systems very fragmented in Europe, they determine whether innovations are actually being picked up. And I try to highlight that too often affordability is treated as a downstream issue, and something that too often is being or taken into account after the innovation already exists. But at that point in time, many key decisions have already been taken and are being locked in. So affordability is not something, again, that you add at the end. It's something you design from the beginning. And bringing affordable and accessible health innovation to patients requires interdisciplinary collaboration on the various levels that affect health innovation, whether they be scientific, regulatory of nature. They require public-private partnerships, an ecosystem that provides risk-tolerant financing, political leadership, and community engagement. And I've asked you to indicate what, according to you, is the biggest challenge. So those who joined a little too late, please go scan this QR code and provide me with your biggest health challenge, or what, according to you, this could be. If you have an other idea that is not covered in the different options given there, don't hesitate to put your answer in the Q&A, and I take it into account at the end of the day to go over how you look at health innovation and the challenges, and of course that those challenges come with opportunities. So the challenges of balancing the sustainability of the healthcare system while at the same time stimulating innovation and still making sure that healthcare is accessible to all is really growing with speed at which technology evolves, with the speed at which drug prices increases, and with the speed at which inequity amongst people between high-income countries and low-income countries, low and middle-income countries increases. And in Module 1, we introduced you to the challenges that come along with this complex R&D process. And there are many incentives to stimulate innovation. And in return for this monopoly position offered through the patent system and the regulatory data protection, the health sector is highly regulated. Professor Bruno Flamion further mentioned the differences between the medical devices and the drug development sector. And we see that regulatory science continues to evolve with, in recent years, also real-world evidence and AI-generated data used to be added to the clinical trials data. As Professor Bruno Flamion stated, the major upcoming impact of AI may be on clinical development. Artificial intelligence is yet to be a game changer in pharma R&D. So far, the anticipated impact is rather limited, but times are changing. And Professor Flamion provided you with an insight in how translational medicine looks like, highlighting some of the evolutions in the field and the challenges industry faces. R&D operating modules continue to experience significant pressures due to, amongst others, the rapid pace of scientific and technological advances and the accompanying ongoing regulatory changes. And during Module 1, you were introduced in the flow of drug discovery from bench to bedside, meaning all the way from the lab to the drug being used by the patients and back. And the relation with regulatory framework for preclinical research, clinical research, translational research, data regulation, and knowledge protection. Now, A challenge here highlighted is, can regulation keep up with science? We saw last week already that regulatory science is key. And we have entered an era of personalized medicine, meaning the right treatment for the right person at the right time. And personalized treatments, including gene editing treatments, are the future, but this obviously implies a rising protocol design complexity and cost of novel biomarkers and diagnostics. During the first module, Professor Flamion took you through the challenging drug development cycle from knowledge generation to clinical application. And for those of you who are not familiar with the drug development sector and the accompanying regulations, don't worry. We realized that we overloaded you maybe with a lot of information at a very high pace. What is important for you after basic lab research and having tested concepts in the lab, we work towards a proof of concept that we will test then in humans. The different steps and phases in clinical trials will be explained to you in a couple of minutes. And although the pharmaceutical R&D process here is presented as a straight line, you know that it is not. I explained this to you. From basic research to improved patient outcome, it is a continuous process of going back and forth, and all different actors in the various phases need common information based on data collection and management, integration, knowledge management, delivery, and presentation. We saw what this course entailed, already providing you with an insight in how translational medicine looks like. We highlighted some of the latest evolutions in the field and the challenges that we faced. But challenges come with opportunities, of course. The age of insular R&D organization is over. To serve a company's innovation engine, R&D strategy needs to be equipped for today's fast-moving world. We touched upon the different steps in the drug development process, and it is a bumpy road. We saw that it's difficult to balance awarding organizations, whether they be public or private. It's difficult to award them IP rights, so intellectual property rights, that could lead to a monopoly position in the market. More and more such patent rights are linked to exuberant prices of the drugs reaching the market. And the question rises then whether we are turning inventions into innovations that add true value for the patients. We briefly touched upon the technology transfer from basic research findings, often spinning from academic research to spin-out companies, or sometimes immediately to big pharmaceutical companies. And Professor Flamion elaborated on the regulatory science and evolution since the last decade, notably during and after the COVID pandemic, and we'll definitely touch upon this during the different modules of the program. So the payer's approval, that is actually what is needed to perform a health technology assessment to inform governments whether or not to reimburse therapies for patients or not. I hope you already had the chance to look at the online course of Matthias Neets on this basics of health economics. This online module is part of the course material. So if you didn't do that yet, don't postpone it until the very last moment. It will give you an insight of what health technology, HTA, health technology assessment, or briefly HTA, is needed, and what do we pay for true innovation? What is society willing to pay? So more and more innovations bring high-priced therapies on the market, and the question is what society is willing to pay for getting access to such therapies, and do they really add significant improvements to the quality of life of patients? How do those innovative drugs weigh upon our health budget? Very, very complex issues, and we need a myriad of experts from different horizons to think about the complexities we increasingly face. I hope this was clear from Module 1. So to answer the question whether we are turning inventions into innovations that add value for the patients, I'm really happy to announce that this afternoon we will discuss value-based healthcare. We have different experts' perspectives reflecting on the prices of drugs and whether we get value for money. So this afternoon we have two people from Novartis who nowadays bring such high-price therapies for rare disease patients on the market. For example, the gene therapy Zolgensma is brought on the market by Novartis, and Zolgensma is the one-shot treatment for SMA or spinal muscular atrophy. And they can potentially get it upon diagnosis, newborn screening of babies. We also have Matthias Neet, so the HTA expert from KCI, the Belgian Knowledge Center Economics, and Lien Beckers, who's currently managing Leuven IDR. And the Institute for Rare Disease, or IRD, sorry. And then patient access. Well, without patients, there's no market, right? And we know already that the place of the patient is right in the middle, right at the center of this whole process. Patients are often viewed as consumers, and although patients often don't have a choice to consume or not, they have preferences. And one patient is not the other, and needs differ. And to this end, so to end this slide, we have the post-marketing surveillance, or the so-called phase 4 trials, where we monitor patients in a real-world setting, and where the newly approved drugs interact with everything that might have been overlooked in a clinical trial setting, such as patient adherence to medication and so on. Again, what is important for you to understand in this whole process is that it's really strictly regulated. With GMP, GLP, and GCP measures that are very closely monitored. Of course, with the safety for the patient in mind. So in the stage of drug discovery, GMP points to good manufacturing practice in place for the early drug discovery in the lab over the whole drug development process. Further, as we move on in the process of preclinical research, good laboratory GLP, good laboratory practice also becomes crucial, with the aim to obtain evidence for safety and toxicity in cells or tissues via in vitro testing and in animals through in vivo tests, and further that we don't sacrifice animals unnecessarily, of course. And then, after having proven safety and toxicity in vitro and in vivo, the first exposure of the new molecular compound can be tested in human subjects. At the time of preclinical success stories, spin-off companies are being set up, and obviously they search for investors to overcome this so-called valley of death, because that's that's when the cost of development really starts to rise. When you want to test a compound in humans during the different clinical trials. Now, this picture here is depicted as a triangle. But actually, it's more like a funnel rather than a triangle. As you know, the attrition rate in pharma is high, with for the 10,000 projects that are being set up, that are entering the pipeline, only one successful on average drug reaches the markets after those 10,000 projects. So high risks, but also high stakes. We are seeing these innovative, here we see the innovative drugs being classified by sales, and the sky clearly is the limit. We discussed the high prices for drugs and the impending and unprecedented sales of the loss of exclusivity of high value assets, the so-called patent cliffs, like Professor Flamion stated. We discussed the rapid pace of scientific and technology advances, and here you see them by already demonstrated medical impact. And Professor Flamion already alluded to the increasing potential of artificial intelligence and machine learning deals in the life science sector. And actually, what you see here with the red line, I don't know whether you actually see my arrow, isomorphic labs. I will come back to that. So the AI techniques will they will alter the way we conduct drug development. It might significantly accelerate preclinical research and reduce the harm we do to animal laboratory animals. We increasingly include new approach methodologies such as organ on chips, 3D tissue cultures called organoids, and computational models such as artificial intelligence systems. They might better mimic human biology than animal models, but are a long way. We're not there yet. We're a long way from replacing animal procedures. And then there's isomorphic labs, for example, because I will always try to allude what I found on the news, I would say. It's a biopharmaceutical spin-off of Google DeepMind. It has unveiled a new powerful artificial intelligence tool for predicting how proteins interact with drugs. The tool, called ISO-DDE, can outperform other AI systems, such as the open source Boltz-2 and physics-based methods at determining binding affinity between a protein and a potential drug. And these skills have really impressed scientists, but they highlight that this system, ISO-DDE, is proprietary, and the technical paper that accompanied its announcement of this system, it offers scant insight into how to achieve similar results. So there it already starts. And we'll see what's coming out of those AI-enabled drugs that entered in the clinical phase the last 3 years already. So particularly the fast-moving AI-enabled technologies can offer hope to many patients facing unmet medical needs. And I take this slide actually of Professor Flamion to highlight, a typo. So there are not 700, but about 7,000 rare diseases where for only about 5 to 10% of the patients face some form of treatment. We'll see much more about rare disease patients today, both this morning and later this afternoon. And in the last 30, 40 years, many incentives have been introduced to incentivize investment in R&D for people facing a rare disease. So facing a high unmet medical needs, such as the European regulation and the US Orphan Drug Act. And it really has improved the situation. A very clear trend in the past years is that pharmas invest in rare diseases. So diseases that are life-threatening, chronically debilitating, and of low prevalence. Low prevalence here means rare diseases affect a small portion of the population, so less than 5 in 10,000 people in the community. So 1/3 of development programs nowadays is targeting rare diseases. The question is where are we with turning those inventions into innovations that add through innovation for true added value for the patients. And you can read more about different regulatory incentives in the articles we put online on the UV, on the virtual system. And I highlight here another article on the European regulatory strategy to support childhood cancer innovation. And by definition, all pediatric cancers are rare diseases. And we'll hear more at the end of this morning from Delphine Heenen, who about what has been done to meet in the past years to meet the needs of this extremely vulnerable patients, because children are not little adults. Skip this for the sake of time. Again, from the news, many policy incentives have been issued, and as I already showed here, those are tackling the regulatory framework that we'll hear much more about later today and throughout the program. And as you probably heard in the news, there's also the Committee for Rare Disease that is being set up in Belgium. And the idea is that it's no longer acceptable that rare disease patients need to wait on average 5 years before getting the right diagnosis. So in Belgium, for example, they will intend to map all different specialties, medical specialties, so that help with or the correct referrals are being accelerated. In personalized treatments, there's the increasing issue of financial barriers. As you know, those personalized treatments come at a very high cost. And advocates asked the FDA to consider approval given the lack of effective treatment options for ALS as the limited signs of toxic effects. They argued that patients had nothing to lose and that potentially even much to gain from having access to the drug. And then financial toxicity, you can ask. The FDA doesn't consider dollar signs was the answer. If the drug discussed had substantial improved patient health, those financial side effects might have been acceptable. But a follow-up trial found that the drug didn't improve the outcomes, and the manufacturer removed it from the market in April 24. How should policymakers consider and address the potential financial toxicity of new drugs in which the balance of health-related benefits and burdens are unclear? Other challenges, quickly to finalize, discussed were the competitiveness from the US and increasingly China, the war for clinical trials, the war on prices, and we will hear more about the most favorable nation tariffs coming from the US. You'll hear more about this this afternoon. And then throughout the course, you'll hear more about the European Pharma Package, the proposal to change the pharmaceutical legislation after 20 years, because it's clear that Europe is lagging behind. So this will be our journey for this afternoon, and we'll hear more about the uncertainty accompanying the market authorization for orphan drugs, which enormously impact drug prices and often targeted a specific patient group nowadays. So here it's really difficult to balance accessibility, affordability for our health system and for patients. And we'll see, and we will definitely hear that the idea is really to accelerate approvals. And this was a statement from Peter Marks from the FDA last year, that it will become the norm, the accelerated approval. We'll discuss the difference between traditional trials, virtual trials, and decentralized trials, and where the concept of real-world evidence and real-world data come in. So this morning you will hear much more about the evolution in clinical research in view of the rise in development of personalized therapies. We'll hear more about the different types of clinical trials, and the randomized clinical trial has been for ages the golden standard, where there are ways to provide earlier access, as I already mentioned or alluded to innovative treatments that meet an unmet medical need. So I will conclude here for this introduction, and I'm very proud with this to introduce to you Dr. Frédéric Hénault, who's a regulatory affairs professional. He has more than 20 years of experience in clinical research and regulatory management within leading academic hospitals and international research organizations. He's the head of regulatory affairs at HUB, the Hôpital Universitaire de Bruxelles, and supports regulatory compliance and clinical trial activities. He has been working for a decade at the clinical trial support unit of the Institut Jules Portet. And before, he worked at EURTC, so the European Organization for Research and Treatment of Cancer. And throughout his career, he has developed strong expertise in clinical trial regulation, project management, contract and budget management, and cross-functional coordination, supporting innovation in oncology and biomedical research. And with this, I hope yes, I managed to stop sharing the slides. And thank you, Dr. Hénault, for being present with us and introducing our students, our participants in the program, into the latest evolutions in clinical trials. Thank you for being with us.
Thank you very much, Hilde, for this short introduction. I'm going to make my presentation, so it's evolution in clinical trial conduct. Uh, I choose to focus it on regulatory affairs instead of having a general overview. You have to know that I will go through a couple of definitions. Many of you may already know them, but I think that it's very important that we are using exactly the same word and same definition because it can lead to misunderstanding when you're dealing with clinical studies and regulatory affairs. So my first introduction will be about clinical trials. And the first very important thing is what is clinical research. So when we are explaining that we are doing clinical research, for example, at the HUB, we are meaning that it's medical research on human subjects. And the aim of a clinical study to find answer to a research question, and we need for that to generate
Just
data.
interrupt. We don't see your slides. I don't know whether you already shared them, but just to know
Oh,
that.
okay. I think I didn't share the screen.
I,
I'm sorry for that.
no, that's not
I'm not
not a
using,
problem. I thought you were still in the introduction of your speech. Now I see myself.
yeah, I'm not using Zoom on a regular basis. We are using Teams. So, do you see my screen?
I see myself now. So yes, now, and if you can, yeah, put it in. Voila, perfect.
Okay. Sorry
Perfect.
for that, Hilde.
No worries, no worries.
So, so my first slides will be about the introduction about clinical trials. So as I said, when we are doing research at HUB and any other public hospital or academic institution, when we do clinical research, it means that we are doing medical research on human subjects. And a clinical study aims to find answer to a question which have been asked by researcher or physician. And for that, we need to generate data for confirming or rejecting the hypotheses which have been made. So the aim of the clinical research is to improving or validating the effectiveness of method for preventing injury or illness. It's also to understand the clinical condition. For example, why do some people develop a cancer? It's also to investigate the cause of the injury or illness. But also there's another very specific field. It's improving or validating diagnostic and treatment method. So we are going to start the definition. So I think it's very important for people to know what we mean by a prospective study because it's the start of the clinical research on human subject. So we mean a prospective study. The timeline starts when you get the positive opinion from an ethics committee. The ethics committee is reviewing your protocol and your formula to get a consent from the patient and they give or not a positive opinion. From the date of this opinion, you can collect data from people who are participating in your research. So it can be data from the medical file, but it can also be also biological samples. To get authorized by the patient to use data and biological sample, you get what we call an informed consent. The patient is receiving a specific form which is describing the study, the aim, objective, and different other logistical aspects, and he needs to sign this document and give his informed consent. So this is what we call prospective because all the data and biological sample that you are collecting are generated after the positive opinion, and you can collect them up to the last patient, last visit. You have to know that in some cases, you're entitled to use data which were already in the medical file or biological sample which were already stored in a biobank. For example, it's the case of Mr. X. So, for example, data that you can use that were already in the medical file would be, for example, the medical history. You want to include patients in your study that had or have a breast cancer. So that's medical history, and this information for sure was present before the positive opinion. There are two types of prospective study, interventional or observational. So in intervention, from a legal point of view, is any action in a study which is not justified by the medical or health management of a given patient. So the definition of intervention is quite broad. It can be a drug, it can be a medical device, surgery, extra milliliters of blood or additional sampling of blood, psychotherapy, diagnostic text, and something very important, which is randomization. So if at least you have one intervention, you have an interventional study. If there is no intervention, we call that a non-interventional study or an observational study. You have to know that people are using observational study in the wrong way most of the time. It's just because they are seeing patient, they are observing the patient, but they didn't take care that they have at least one intervention, which is driven by their study protocol, making it an interventional study. So I really want to emphasize that this definition is very important when you're discussing with project manager, with pharma companies, or even with regulatory affairs. You really have to be careful about that. If you go to the prospective interventional studies, they are quite broad. It's a very huge family. And there are three types of studies that you really need to clearly define and use the correct name for them, again, to avoid misunderstanding with regulatory affairs. So when a prospective interventional study is called a clinical trial, it means that a medicinal product is the object of your study. If you're discussing a clinical investigation, a medical device is the object of the study. And if you're discussing a performance study, it means that an in vitro diagnostic medical device is the object of the study. So I really emphasize that you really need to use this word and these correct names for your study. Something that you have to be cautioned. Clinical trial has a different meaning for the World Health Organization and for some other countries' entities. So I'm using here the definition that we are using within the European Union. So again, check where the people are from. If you're interacting with people from US, they may misunderstand what you're meaning by clinical trial. So in that case, just tell them that the medical product is the object of your study. So there's different phases in clinical research. First, you have what we call preclinics. It's analyses and hypotheses which are, made and verified in laboratories. And then it's what we call the clinic phase, is where you're having patient or subject. Your clinical trials are defined in three different phases: phase one, phase two, phase three. Then you get the marketing authorization for your medical drug. And then you have what we call the post-marketing follow-up. And most of the time, these are called phase four studies or phase four clinical trial. So which are the differences between all these four phases? For phase one, what's very important is that the sample size is very low. You have about something like 30 subjects, and you are interested about safety and tolerability. So you try to figure out which is the best dose, which is given the less side effect to your patient, or which is giving some effect. Then you move to phase two. It's a medium-sized sample, 100 to 300 subjects. You are studying the side effect and their management, and you're also looking now at the efficacy as the expected result produced. Once you have a phase two with positive result, you move to a phase three studies or trial. It's a very large sample size. Most of the time, you have more than 1,000 subjects. It's run worldwide. You have randomization. And most of the time, you're comparing the new treatment with the clinical routine practice. Once again, you are checking efficacy and safety. If you move to phase four, it's a real-life use after market introduction. You are checking the common and rare side effect, and you see how your medicine is comparing to alternative treatment. You have to know that phase four studies may be mandatory depending on the type of the drug that you are dealing with. For example, in oncology, most of the drug need to go through phase four, and it's a requirement by the European Medicine Agency. So if you want to run your clinical trial, well, it's a very complex organization from an operational point of view. So you start from your research project, and then you have to go through all these different steps, which make it look like a clinical trial roadmap, meaning that regulatory affairs, it's one of the pivot, because without any approval, you cannot go to your clinics. I'm going now to discuss the evolution of the legal framework regarding to research. So first, we have an ethics framework, and it started a long time ago with the Hippocratic Oath. And this oath is establishing several principles of medical ethics, which remain of paramount significance today. Most of the ethics framework is a response to the human abuses, for example, during the Second World War, where experiments were made on human beings without them being informed and or having given a consent. You have a different type of document. The first one is the Nuremberg Code. You need to know that it's the start of this ethics framework after this World War II experiment. And two documents are very important for you when you're doing clinical research is the Helsinki Declaration and the recommendation of the ICH, and it's what we call the good clinical practice, the GCP. So if we go deeper in the legal framework, you have to know that when we are asking for authorization or opinion to run a study, whether a clinical trial or a clinical investigation, we are subject to the legal framework, and we have the laws, which is a rule made by an authority and that you must obey. We have the directive. A directive is laying down certain results that must be achieved, but you don't know how you can achieve this result. It's up to the country to decide the best way. And it means that individual countries need to create or adapt their national legislation to reach this goal. And they do it by having laws. And then we have what we call the regulation. And the regulation is a rule or order which have been issued by an executive authority or a regulatory agency, and it's having the force of the law. So it means that you don't need to transpose it into the national law. It applies automatically to all the EU countries as soon as it enters in force. But you have to know that because of this regulation, you may have to modify your national legislation. And once again, you do it through laws. There's also other different legal framework that we need to follow when we are running clinical studies. I'm not going to go into details for all of this, but you have to know that they exist, and sometimes we are making references to them. You have Act of Parliament, ordinance, decrees, administrative circulars, and sometimes guidelines can be also binding. So if we go a bit deeper, we are going to discuss the good clinical practice because it's something that every single researcher doing research on human beings need to know, and you have to get a certificate which is validating your training. So in 1996, it's the first version of the ICH GCP, and then it's focusing on credible and accurate data and protection of subject rights, safety, and well-being. In 2016, it has been modified, and we have introduced a risk-based approach for quality management and monitoring, and use of technological tools like the electronic trial master file, the electronic data CRF, and certified copy. And most recently, we have the third version in 2025. And now it's imposing practical feasible expectation through adoption of quality by design and risk approach. And they are also embracing technology advancement, addressing data acquisition with electronic tools. If we go more in depth for the legal framework for clinical trials, we move from a clinical directive to a clinical trial regulation. So in the past, you were supposed to obtain the authorization to run your clinical trials according to national rules in each EU countries. Uh, the problem was that at that time you didn't have any legal framework which were making it very easy because every national authority has its own rule. So in 24, we had the Clinical Trial Directive, which is the Clinical Trial Directive 2001-20 EC, and it has been implemented in the Belgium law and it's the law of the 7th of May 2004. So at that time, the directive was imposing for clinical trials that you had to get authorization from a competent authority, opinion from an ethics committee to run your clinical trial within the member state. Is also having imposed other obligation. For example, you have to publish the result of your clinical trial. The problem is that because it was a directive, it has been interpreted differently by some lawyers in different countries. So, for example, in Belgium, you're supposed to get the authorization of all the local ethics committee and also from a committee which is responsible for a unique opinion. And in some other countries, there's only one committee which is responsible for this opinion. It's the case, for example, of Portugal. It's a national committee. And, or for example, France, where an ethics committee is regional and is giving his opinion for multiple centers. So even if it simplifies the process for international trials, we still had to go through national legislation and differences were quite huge. So it took 10 years for the legislators to go to a full harmonization, and it's where we have the clinical trial regulations, which is called the CTR 536/2014, and it has been implemented in the Belgium law of the 7th of May 2017. So, as I explained before, this clinical trial regulation was immediately applicable to us, but we had to modify our legislation through this law of 2017, and it has been modifying the law, which has been almost the same date, but from 24. Between 2014 and 2022, there was a pilot phase run by some countries, for example, Belgium and France, to allow the legislators, the authorities, the ethics committees, and the sponsor to get used to these new processes. And it has been fully entered into application in 2022. You have to know that from 2023, the clinical trial regulation is mandatory for all new submission of clinical trials, and we are now in the full application of the clinical trial regulation. So what does this mean for us when we are under this clinical trial regulation? There are key features. For example, we have identical rules for conducting clinical trials through the European Union. We have a single submission and approval of multinational clinical trial applications through an electronic platform, which is the Clinical Trial Information CETIS. Co-sponsorship is allowed. It means that you can have one or more sponsor. So when I'm using sponsor here, I'm using the legal definition. It means that it's a person, entity, or any other groups or institution which is taking the full responsibility for the studies. Is not the entity or company which is bringing in financing and or, for example, goodies. So you have to be careful when we are using sponsor. It can mislead people. There's also an harmonized procedure for assessment by the member state, and it's divided into parts. We'll go to that on the next slide. There are strictly defined deadlines for the assessment, and they're applicable to sponsor, competence authorities, ethics committees. We cannot get any waiver. So just to give you an example, when you are receiving question during the assessment of your dossier, you have 12 calendar days to reply. If you reply within 13 calendar days, your application is rejected by the system and authorities don't have any control on that. So it means that you get a disapproval. There's also mandatory reporting of serious breaches through the clinical trial information system. We were supposed to do it in the past, but people were not doing it, or it was not clear depending on the national laws if you were supposed to or not. There's a definition of a low interventional clinical trial. I'm not going to go into details. You just have to know that it's something which is new, but not really applicable to Belgium. We didn't get any advantage from that definition. There's a strong transparency of clinical trial through the publication of all the studies. Also, in these cities, you have to publish all your results. And something very important for the worldwide community is that the public has access to the detailed information on all clinical trials conducted in the EU through a public website. And you can get all this information once applications are submitting and approved. So the sponsor need to upload redacted document as well as unredacted document to protect data privacy and intellectual property. So if you want to know about a protocol which is run by GSK, you can go to CETIS and see and get the protocol. So what's very new for us, it's the way that the dossier is assessed, and there's two parts of the assessment. First, you have a part one, which includes the assessment of the protocol, the investigator brochure. Investigator brochure is giving you all the relevant detail about the drug. The investigational medicinal product dossier, mainly it's explaining how your drug is manufactured. And assessment of this document is done within EU mandating timelines and is led by a reporting member state, which is coordinating the assessment with other member states who are participating to your trial. There's a part two of the dossier, and this is assessed by the National Ethics Committee, and it includes the assessment of the subject information and informed consent document, the suitability of the investigator and clinical trial site, indemnity and data protection. Oops, sorry. So each member state will consider the conclusion on part one, the conclusion on part two, and then you get for each country either an authorization or an authorization with condition or disapproval. And all this information is found in CETIS. So, as I explained, the legal framework for clinical trials is very complex. So, if you're just focusing on Belgium, we are under 5 regulations: the clinical trial regulation, a regulation about the GCP inspection, a regulation for the good manufacturing practices of the medicinal product and the related inspection, a regulation which is detailing the safety assessment, and the last regulation was about labeling of the investigational medicinal product and the auxiliary medicinal product. We have one law, which is the law of the 7th of May 2017, and we have also two royal decrees. One is related directly to the clinical trial regulation, and one is related to the Federal Agency for Nuclear Control. You have to know that in Belgium, if your investigational medicinal product is a radiopharmaceutical, you need to get also a specific authorization from FONC before running your trial. And just to add an extra layer of complexity, if you have medical device in your clinical trial, you're also subject to the medical device regulation, the 745 and or the 746, and associated laws and decrees. This regulation may apply, and in that case, it's what we call combined studies. So if we move to the clinical trials in Europe, well, Belgium is doing quite well because as you see that the evolution over time per 1 million capita, Belgium is a very good country for running clinical trials. Belgium is also having a lot of clinical trials from phase one compared to the average that we have within the European Union. So the explanation behind that is that our legislation was very favorable for this type of study. And if you check the proportion of clinical trial application for a selection disease area in Belgium, you see that most of them are run for cancer research and also nervous system disease and immune system disease. The evolution of the clinical trial application for cancer in Belgium have been quite steady over time, and we had a small decrease, which was because of the COVID crisis and also with the transition from the CTD to the CTR, but it remained quite steady. So if we move to the challenges that we are encountering as regulatory affairs, but also as sponsor of clinical trials, well, the average rate of Belgium for the following drivers are checked, and we are going to concentrate first on startup timeline. As you can see that from 2017 up to now, the startup timelines are decreased on this scheme. It means that they are very bad for us. So why is that bad? And people were wondering. First of all, we have a lot of competitive for Belgium. Under the EU Directive 2001/20 EC, for a monocentric phase 1 trial, it was taking up to 45 calendar days to get your authorization. Under the CTR, we go up to 66. For other trial, the max calendar days was 58, and under the clinical trial regulation, we go to 106 calendar days. So our competition regarding to timelines is not good anymore. So because of that, sponsor authorities have been discussing, and now there are new timelines, new processes, and new regulations. So this is fairly new. For example, there are new timelines in Belgium since the 1st of January of this year. So for mononational phase 1, 1, 2, and 2, you go up to 59 calendar days, sorry. For mononational phase 2, 3, 3, 3, 4, and 4, it will be max 79 calendar days. Just as a reminder, under the CTR, it's 106. The only issue that we are encountering is that these new timelines are not binding. They are not set in by the law. So it means that if the authorities and the ethics committee cannot keep up with that timelines, you cannot raise complaint. Belgium also agreed to be part of the FAST-EU. So this is for international clinical trial. It has been launched by the European Medicine Agency and the European Commission in January of this year, and it's aimed for an overall duration of 70 calendar days from submission to conclusion, including sponsor response timelines for all these international trials. Pilot phase has already started. They are accepting only 2 trials per month, and it's a voluntary-based participation of the sponsor. And the last initiative, it's a proposal for a Biotech Act, and this has been published on the 16th of December 2025. This is an initiative launched by the European Commission, and it's concerning innovative health innovation and product, for example, CAR T cells, antibodies, and they are aiming at shortened timelines for clinical trial, meets up to 47 days without request for information. Currently, there will have further consultation and legislative development ongoing through this year to refine the framework. So if we go to other driver for clinical trials in Belgium, you see that adoption of new technology didn't improve. And it's kind of a pity because there are a lot of new technologies that are developed and you can find a lot of publication now about them. For example, you have a personalized medicine, you have targeting a specific type of cancer, you are using radiopharmaceutical products to help patients going through their disease. You have progress of mRNA vaccine and immunotherapy. But also you have, for example, hypnosis, where it can be interesting for some patients. You have the use of artificial intelligence in healthcare. You have ePro-based individual follow-up. or otherwise you have also other type of RPO. So Belgium really need to keep up with the adoption of these new technologies. And if we go to patient recruitment efficiency and education of population of clinical trials, you see that we are not also very good for these two areas. So if you check the timelines for patient recruitment, for example, in oncology, in most of the countries, you see that it takes 1 to 5 months to get the first patient in, but for some countries, it can go up to 2 years. Rare disease, Belgium is doing quite well. And for infectious disease, you see that the timelines are more spread over time. But to get your first patient in within less than a year, it's of tremendous importance to run your studies. And you see that we really need to work on that. How can we make the recruitment faster? Well, to make the recruitment faster, it means that you need to empower the patient in clinical trials. So the patient is an equally important stakeholder. You need to make them aware and give them some education. You need to provide the information about your clinical trials. So it can be upstream informed consent or downstream. It means that you are publishing the clinical trial result. And something which is new under the clinical trial regulation is that you may need to have the patient participating in the design of your study. It's one of the questions which is always asked. Does the patient have been involved in the design of your clinical trial? So there's this European Patient Academy on Therapeutic Innovation, EPATI, which is a worldwide association for patients. And recently, Belgium launched a website, clinicaltrial.be, where you can find information about the clinical trials which are run in Belgium. So it means that the patient can be proactive and do themselves some of the research if any trial is corresponding to their area of disease. So I'm going to finish my presentation, so it will be the take-home message. Well, where clinical trials take place matter, and their value is quite extended beyond the benefits of patient. So indeed, first of all, if a patient is participating in a clinical trial, he may have benefits. You cannot guarantee that the drug will cure the cancer of this patient, but the patient may have benefits. There are benefits for the healthcare system because clinical trials bring revenues, are cost saving, they improve clinical outcome, healthcare performance, and also it's very good for the society itself because it improves the health and well-being of the society as a whole, and it can bring also economic investment in Europe. So when you want to run clinical research, but it's a collaboration between all stakeholders and it's essential. You have the authorities, hospital, patient, universities, and industry. Thank you very much for your attention.
Thank you. Thank you so much, Dr. Reno, for this very enlightening presentation, particularly indeed in light of the various reforms that are being proposed. If there are questions from students, don't hesitate to put them in the Q&A. If you don't feel comfortable asking them yourself, I can see them appearing and I can ask them for you. Maybe because you alluded to this FAST EU and the Biotech Act, can I ask you with respect to AI-enabled clinical trials? Professor Flamion 2 weeks ago alluded that there are some trials based upon artificial intelligence and machine learning are now, well, the last 2-3 years being entering this whole pipeline, this clinical trial pipeline. What is your experience? What do you expect? What do you see as challenges and opportunities? Big question, sorry. So Please go ahead.
Yeah, no, it's okay. I mean, so yeah, you have to know that people are running studies on AI, for example, to develop diagnostic tools or tools to help you to make a diagnosis. So most of these development are covered by the two medical device regulation, because if you have a software which is making a diagnostic, it's a medical device. So it can be either under the medical device regulation 745 or the 746 if it's an in vitro diagnostic medical device. So it's already covered by these two regulations, but you have to know that there's the AI Act, which is entering into force, and it's going to be this year. I would say that nobody's having clear experience with that. It's a black box, and we are going to be subject to an additional regulation. And it's what people are afraid of. That, for example, if you have AI within a clinical trial, it means that you'll be subject to three different regulations, which sometimes are going in two different ways. Just to give you an example, if you're subject to the clinical trial regulation and one of the medical device regulation, in some cases, you have two different independent ethics committees which are reviewing your study. So it
Incredible.
means that you get different opinion, you get different comment, and they're not consolidated. So it means that you need to go back and forth between all these entities. And these have never been foreseen by the legislators. So I know that there's something which is done at the EU level where they are trying to put a pilot phase for reviewing this type of combined studies and come up with solution for sponsor.
Yeah. And there's a lot ongoing also for orphan medical devices, because this is also like a void or a gap in the regulatory system well, currently under the medical device regulation, but there's no real addressing the need for more orphan medical devices and the sustainability in the production and the maintenance, particularly when it comes on the market. My interesting. And can you tell something more about this regulatory sandboxing with respect to like loosening up the regulatory system and control making or creating controlled frameworks where the control is a little bit loosened? Do you have experience there? Because this is, if I'm not mistaken, included in the Biotech Act.
Yeah, so we don't have any experience because the Biotech Act is only a proposal so far. So we need to see if it will be adopted or not. But in fact, it's very interesting for all kind of sponsors that they are losing the tide, because it may help us to go and get faster approvals and dossiers which are not that complex. So we are
Yeah.
crossing our finger that it will go. If people are interested, the draft of the Biotech Act is available on the European Commission website.
Yeah, I will indeed make it available. I see, Ghana, you have a question, but I don't see your question, so I hope. Yes, you are there. Please unmute yourself. Hello.
Hello, thank you for
That's
letting
great.
me speak. Thank you, Frederick, for the presentation. My question is, as a new professional, let's say in the clinical trials, if you would like to pursue regulatory affair course or direction, how would you begin? Where would you get an internship in that field? Thank you.
If you need to go into regulatory affairs, there's a different organization which are providing training and certification. I'm not going to give the name just not to make publicity for them, but you have to know that you can get certification from different organizations, even in Belgium. And from that, you get experience directly in the field when you can be hired by companies or academia.
For example, I just completed the clinics program with the ULB, but there's no specific section, let's say, for regulatory affairs. So if I'm interested in transition into regulatory affairs, is it possible to get an internship in this field?
Yes, I mean, you can either go and get, for example, training at the HUB of CTC. I mean, we are collaborating with clinics, for example, and we have trainees who are coming to our department. You
Is
can
it
get also trainings by going to this organization, which are specialized, and they're advertising their training on LinkedIn, for example. So there are different means of getting it, and sometimes you just need to have a legal background or a, I would say a scientific background, and some companies are just hiring you and they're training you.
is it possible to send you an application for a possible internship?
Oh, yes, sure, it's always possible.
Uh, okay, so maybe perhaps I will get a contact from Madame Stevens.
We can definitely arrange this, and
Thank you.
definitely indeed.
Thank you very much.
You would not be the first program participant to enter into the whole system at HUB. So definitely this is possible. So do you have other questions?
No, thank you.
Okay. If there are no other questions from the students, I would like to thank you, Dr. Hinault, for this very interesting presentation. I think there is a lot coming to us. It's very interesting and exciting to feel that the field is moving, that we indeed are running against the wall of regulations. It's clear that Europe and Belgium is lagging behind, actually, and that there are, as I explained to the students in the introduction, there are many, many, many things ongoing to help improve the competitiveness of Europe in the field, particularly with respect to the US and also and increasingly China. The fragmentation that we are, I would say, subject to in Europe is a hurdle. Luckily, I would say for clinical trial regulation, it is better than for the pricing and reimbursement. That's even worse. This we will discuss in the afternoon. So thank you so much for this
You're
interesting
welcome.
presentation. And then for the students, we will reconvene at 10:45 for the presentation given by Geoffrey Istas, and then afterwards by Delphine Hien. Thank you so much.
Thank you,
Dr.
Hilde.
Thank
Bye.
you.
Thank you. Bye. And for those who are still there and who haven't participated in this WooClap event, please don't hesitate to scan the code and check what you think is the biggest health challenge. See you at 10:45. Bye. It's indeed 10:45. Thank you for being back with us. I see that in the meantime, 101 people entered this WooClap system to answer this question of what, according to you, is the biggest health challenges, but only health challenge, but only 87 people answered. So I'm wondering what the other 14 are hesitating about. Maybe they think they are all challenges or they don't see it as a challenge. I'm curious. And also for those 3 people who answered that they had something else in mind, please do not hesitate to provide us with your answer or your thoughts in the chat, in the Q&A here in the Zoom, because I would love to discuss at the end of the day, how you see it or how I see it or how the speaker saw it. So very interesting, and thank you for participating. And with this, we will start with our next speaker, Dr. Geoffrey Istas, who's a biomedical scientist with a PhD in cardiovascular and nutritional sciences, jointly conducted at King's College London and the University of Düsseldorf in 21. He further expanded his expertise through an external education program at Harvard Medical School on novel therapies for chronic inflammation, immunity, and allergy. This interdisciplinary profile bridges molecular biology, genetics, bioinformatics, and translational health research, supporting innovative approaches at the interface of nutrition, epigenetics, and disease prevention. I'm very, very happy because Dr. Istas will bring us an insight into how we can bring innovative therapies to patients. Thank you, Dr. Istas. The floor is yours.
Thank you very much. Can you hear me well?
Yes, we hear you.
Yes. All right. Good. Thank you for that nice introduction. So, yeah, it's my pleasure to give you some insights on what's cooking at argenx and how we develop our therapies. So, let me share my screen. Okay.
Yes.
And then presenting mode.
Yeah, perfect.
All good. All right. So, yes, I'll move straight to what I'm going to talk about today. So it's really my pleasure to give this talk and give you some insights on what we're doing and maybe inspire you as well for some of the next steps in your careers. So I will briefly explain who argenx is, who we are, what we do, where we come from. And then move to how we develop novel antibodies, and then give some examples on innovative trial designs we've been working on, and that also helped us to bring these therapies to actual patients, and finalize with some key takeaways. So what I propose, and Hilde, let me know if you agree, is that after every section, I can pause a little bit to see whether there are questions. Yeah. All right.
Perfect.
And then based on that time, I will see whether I skip some slides or not to make sure
Okay.
we stay in the time.
Perfect.
Yes. So how it all originates? Well, basically it's three very ambitious and smart people that came together back in 2008 based on an idea to innovate in autoimmune diseases and create the next breakthrough immunology therapies. And that was based on an insight from one of the founders, Hans de Haart, who saw that llama-based antibodies are very human-like, which allows to skip some steps in the development of antibodies and go fairly quickly in that development process. And based on that idea, and that, massively, how you say, developments and knowledge on antibody engineering, that the company was built. But it's not only based on that, and that's what this slide shows. It's also based on a very specific culture that was instilled from the beginning on how the company should function and how people working at the company should behave in a very specific way. And this is also part of the key to success at argenx, is how, the culture of argenx. And that's where the name comes from. And actually, it's based on Greek mythology. It's a story of Jason and the Argonauts. And that was basically the first written story where not only an individual person excellence was put in the picture, but a team of people achieving the unthinkable. And so it's rather the individual contribution of special team members that made a collective effort reaching the unthinkable. And that is exactly what argenx is about. It's more teamwork and empowerment to achieve the unthinkable. I just wanted to mention that. That's really part of our culture. And so what we do is we really bring science and innovation to patients, right? That's really our core mission. And this is basically some of the important cultural behaviors and pillars that we're about. They're really not just words on a wall. It's really something that is very intentionally created and invested in today as well. And there's a few things that stand out here. So humility, empowerment, and co-creation really enable this innovation and excellence that is happening at argenx. Now, having said this, how do we move and how do we develop novel therapies, right? So that's really based on our immunology platform, so on our expertise in antibody engineering. And we partner, so that's also key to success. And where the humility comes back is that we don't do it all ourselves. We don't pretend that we know it all ourselves. We are good at creating antibodies, but we need help from lead academic experts that spend their lives working on a biological pathway, on a disease indication, to give us insights on what are the good targets, what are the targets that make sense to develop a therapy that is also really transformational for patients. And that partnership is really key to success because they give us these biology insights which we would not be able to understand ourselves. What we do very well is creating the therapy against a certain target. That's something we can do very well. And that's what highlighted here. So as I said before, right, the llamas are a key part of this story. We developed this platform based on llama antibodies. It's called the Simple platform. And it allows us, as you can see on the bottom of the screen, it allows us to develop from the idea of understanding what the target should be in the body. It allows us to develop an antibody in 3 months' time already. So that is really exceptionally fast compared to sometimes it can take years for conventional other ways of creating antibodies. And one of the reasons I already explained, it's because they're very human-like, the llama antibodies. Even though we don't look alike, the antibodies, and especially the variable region in this fork-like structure, so the upper part of the antibody. Let me take my my pointer to help. So the upper part of the antibody here is very, it's the piece that binds to targets, to specific targets, that is very human-like. And so it allows us to skip some steps in making sure to be able to inject in humans. An additional benefit of these antibodies from llamas is the llamas that we use. So first of all, they're very happy outbred llamas living on a farm somewhere in France. And what is really essential is that they are outbred. So whenever two llamas make an offspring, the two llamas must be as far as possible from each other in terms of relative. So it's not brothers and sisters, but really distinct species of llamas as much as possible to create a very variable gene pool and also antibody pool. And it's also that aspect that allows us to basically create an antibody against any target in humans, but also in rats or mice or monkeys. And the latter also helps us with only one platform to not only do clinical development, but also, of course, the preclinical development to understand before we inject in a human, well, does the biology work? What are potential side effects? We can do that all with the same platform. So that's basically the variable piece of the antibody that has a lot of benefits in our system. But then there is also the what we call the constant piece, the piece that never changes in the antibody, the Fc region. And there we've also done a lot of engineering, which I will explain a little bit later, that gives us some advantages compared to other therapies. And this slide showcases what we then call the IIP. So it's the immunology innovation platform. And that is really what I was saying, that we're bringing the antibody expertise, but it's more the partnership and the co-creation with these academic lead experts to find the targets that allows us to create all these molecules that we're doing. And we don't just partner with them at the beginning to have the idea and then and then move forward without them. No, we bring them along the way, along the entire clinical development way. And so they keep on feeding us with important information, these experts, throughout the whole development of the compound. That's also an essential piece. So these are some of the key compounds or molecules we have in-house, right? And the first from the left part, I will explain a little bit. Well, maybe I'll pause here actually. Now that I'm thinking, I don't know if there are already questions. No?
No.
All right, great. Then I'll continue. So I'll give a little bit of insights on which type of molecules we're working on and how far they are in the pipeline. And I will cover these three ones in more details. So first of all, our lead molecule, it's also the one that is on the market. It's the only molecule we have on the market so far, is efgartigimod. And just looking at the time, yeah. And efgartigimod is really what is driving also Argenix success so far, because it's making us also an independent functional company because of commercialization and its success. And efgartigimod is actually a piece of an antibody. It's not a full-length antibody like you would see here. It's only a fragment. And it's designed to target a receptor, and it's called a FcRn receptor. And that antibody was also developed in collaboration with Dr. Sally Ward, that knew everything about FcRn. She was the one telling us, well, it would be interesting to develop a therapy because it can have a huge impact in autoimmune diseases. And she was also the one to say, well, use a fragment rather than a full-length antibody because of various reasons. And we see up until today, compared to competitors, why that has benefits in, for example, side effects and these kind of things. Again, highlighting how important it is to really stay humble and say, we don't know it all. Give us any insights you have, and we partner together to a solution. This is how efgartigimod. So this is how. Yeah. Now I'm going to explain a little bit how efgartigimod works. But before I go there, I want to understand the norm. I want to make sure you understand the normal conditions. So efgartigimod targets antibodies. So it's strange, right? It's an antibody therapeutic that targets actually antibodies in humans. And what it does, it reduces specific type of antibodies, which we call IgG. They're the most predominant antibodies. And they reduce, efgartigimod reduces the IgG levels in the blood. And what happens in many autoimmune diseases is that we have our own IgG antibodies that are attacking our own body, or some organs. And so then you want to reduce that in that setting. And so what happens, how can we reduce IgGs? Well, IgGs, they use in normal conditions, a recycling pathway, and that's that FcRn receptor, to extend the half-life in the blood. So making sure that these antibodies can remain for weeks, up to 3 weeks in the blood, as compared to other proteins that don't have that luxury to be able to recycle by cells. And that's what is shown here. I'm not going to go into too much details, but what it does, it's when it goes in circulation in the blue piece, it can then enter in the cell through a natural process of internalization. And in the cell, the first piece here that you see is called an endosome. And that is where the antibodies and everything actually from the plasma goes in that endosome. And the cell then starts to recycle or break down proteins. And that is by reducing the pH levels and having some proteins and enzymes that break down the other proteins. But what rescues these blue antibodies, these IgG antibodies, is their binding to FcRn. When they're bound to FcRn, they are actually rescued from being degraded in lysosomes, which is called a lysosomal degradation pathway. And so by being bound to FcRn, they're kind of protected, and they can get back to the surface and then be recycled. And that's like a cycle that allows these blue antibodies to remain for a long time in the plasma. What efgartigimod does, it blocks that binding to FcRn. So it outcompetes these endogenous IgGs, these endogenous antibodies, that then suddenly cannot bind anymore and be protected by FcRn. And so they go also in that degradation pathway, and that's how we reduce IgG levels in the plasma. And so again, the aim is, well, in autoimmune diseases where IgGs are a problem, of course, it makes sense to reduce them. And that's how we've been managing to show that this antibody fragment, this therapy works in, for example, myasthenia gravis. It's a neuromuscular autoimmune disease. In CIDP, so chronic inflammatory demyelinating polyneuropathy, it's also a neuro disease where antibodies destroy the nerves, the peripheral nerves. There, again, we show that by reducing these antibody levels, patients get better. And then in as a third one that also has shown, and we're on the market also for this indication, is in a blood disease called immune thrombocytopenia, where actually the patient's own antibodies are destroying platelets. And platelets are important for clotting and whole blood cell homeostasis. And then you see bleedings and hemorrhagic blisters, and it's all these diseases are horrible actually. They're fairly rare and autoimmune, and that's exactly what argenx targets. Second one is the C2 blocker. So that's another compound that is not on the market yet, but we're in full development. And that is using something that we call sweeping technology, sweeping antibody. What comes back here is that FcRn receptor, right? That is in natural conditions there to recycle antibodies. Well, the sweeping antibodies will also bind and outcompete this the endogenous antibodies to bind preferably to FcRn. But what it will do is that you can design an antibody that targets a specific protein in the plasma. And so it stays bound. As you can see, the black little bullets here are, for instance, your target or your pathogenic target in the plasma. It binds to that. It will be internalized through FcRn. So it takes away these black bullets from the plasma. However, with reduced pH in this endosome, in these little compartments in the cell, it will then release the target. So the black balls are then released in these little compartments, which are then, of course, go to the degradation, and they're then degraded by the cell, while the antibody remains bound to FcRn and gets recycled again and can do that again. And that's why we call it a sweeper, because it sweeps the target away from circulation to bring it to a degradation in the cell. And that's a way to very selectively remove a pathogenic protein from the plasma. And that's exactly what we've been doing. This is also in collaboration with some professors that know everything about complements. And they propose, if you want to target the complement system, which is an important pathway in immunity, you can target C2, which is a factor in that complement pathway, and use that sweeping antibody to reduce C2 levels. I see there's a hand up. I don't know. You can ask your question if you want to.
Yes, Julie, go ahead.
Yes, I was wondering, because you mentioned that you use an innovative way to develop your antibodies, and that you can develop an antibody in 3 months, and that it's a lot faster compared to other companies. Does that also mean that you can lower your price because you can go to the clinical trials and stuff much faster than other companies?
Good question.
Yeah, that's a good question. Yes, I think in the pricing setting, and I'm not the expert there, I'm just a scientist, but it will help to have price negotiations with payers, because you can go faster in timelines. But it's still, the 3 months is only a piece of the story, right? It still took efgartigimod, I think, it still took around 8 years to go from one to the other. And the pricing, I mean, I'm sure you'll have other sessions on pricings, but it's much more complex than just the timelines of clinical development.
Yeah.
But what I can say is that our price negotiators, they at Argenix, they use like a value-based type of system to negotiate prices. And so they really look at what is the additional value that our product can bring to make a difference to justify a certain price. But other than that, I cannot comment much more.
No, we will. But thank you, Jacques,
Mhm.
for your answer already so far. And thank you, Julie, for the very interesting and insightful question, because you already show that you are able to bring pieces together from the things that we will indeed discuss more on this afternoon, actually. Thank you.
Okay, thank you.
Yeah. So yeah, that's so back to that molecule. So that complement sweeper, right? Here we show that using this antibody targeting C2, it can really reduce C2 levels. And actually, yeah, the curve on the right here shows the normal circulation time of a normal antibody. So it's around 3 weeks, a little bit more, sometimes a month. But by tweaking some domains in the antibody structure, we can increase the half-life. So that's another piece of engineering that we've implemented, and increases actually the circulation time by being better bound to that FcRn receptor and recycling better. It allows to stay longer in the blood, and so the impact of that is that you may dose less frequently, right? That you don't need every week or every 2-week dosing, but only every month, for example, or every 2 months, depending on what the disease is. And these are some results of a phase 2 trial in also a motor neuropathy, and that showed a clear difference with patients receiving placebo versus our product, where you saw that the probability of being rescued with a therapy with a standard of care was much more different between placebo and between the gray curve, which is the placebo, versus the blue curve, which is our therapy. So patients stayed much more stable on the therapy compared to placebo. I saw a hand up, but it disappeared again.
Yes, excuse me, Geoffrey.
Yeah.
I have a quick question. Do you develop something against the complement 3A receptor?
No. The complement pathway that we target is C2,
Okay,
the C2
thanks.
factor. Yeah.
Sure.
There are also, I can speak to that a little bit. So there are some advantages of going to C2 versus later cascades of the complement, is because you have like three pathways in the complement, right? The alternative, the lectin, and then the third one, which escapes my
Classical.
the classical pathway. Thanks. And there, by going to C2, you can keep you can target 2 pathways and keep one intact. And this allows to reduce the inflammation or the overimmune reaction, while still keeping some pathway against kill against bacteria or natural infections still intact. So that is a little bit the balance that for example, a C2 blocker can give, as opposed to if you go later down in the cascade, like a C5 inhibitor, it shuts down all 3 complement pathways, and then you don't potentially have less natural protection against.
But just a question, I mean, you have C3A and C5A receptors on macrophages. Why not target those rather than, classical and lectin?
I think on macrophages, so macrophages are not the only cells, right? So I think by targeting in this case C2, for example, you can also target C1. It targets a broader population of cells, of immune cells, or a broader impact on the inflammation caused by the complement. But I'm not a complement expert,
Oh, sure,
I must say.
But
Yeah.
do you have any ways to, let's say, target specific complement? Like, there's this new idea that there's intracellular complement, for example, that is specific to certain organs, or you just
For now, I'm not involved in those discussions, so I don't know, to be honest. But it's a good question. But I don't think so, that we're looking at that for the moment. No. All right. The third one is the last one I just wanted to highlight. Let's see. Yeah, there's still some time. Is another asset, another antibody that we've developed in collaboration with some experts, and it's a MuSK agonist. And what is a MuSK agonist? So the MuSK receptor is also very important in the neuromuscular junction, in the normal functioning also of the neuromuscular junction. So knockout mice that don't have MuSK or any components linked to the MuSK receptor, well, they're not even, well, they die very early. Some are not viable because it's such an important piece in the creation of this neuromuscular junction and having motor neurons and muscle working in the body. So what what was interesting in this collaboration was the idea by the experts to not develop an antibody that would block something, a blocker receptor, but more an antibody that would be used as an agonist. And an agonist is actually have an activating function by binding to it. So it's a bit of an opposite way of thinking. And here, what is very interesting is that, so in biology, MuSK receptor, you need to have two of them brought together, like physically brought together. And that's what happens in a healthy condition. You have intracellularly, you have these DOC7 proteins that bind to several MuSK receptors, and they dimerize the receptor, and by dimerizing only, the whole cascade can function, and neuromuscular junction is established. And here the idea was to use the two arms of the antibody to dimerize the MuSK, as simple as that, actually. But you need to be able to think about that, right? To have that idea. And that's exactly what they did. And we developed the antibody that binds to a certain region of the MuSK receptor. And by doing so with the two arms, it can just physically bring the receptor together. And then the idea is in diseases where that process of dimerizing is not happening because of some mutations or whatever, well, maybe the antibody can help and make the difference. And that's exactly what we've seen. Still very early in development. It was more of a proof of concept setting, where, so not even a phase 2 yet, but we've got some results in congenital myasthenic syndrome, where in those patients, they have a problem with the MuSK and the neuromuscular junction. It's a very severe disease. They have it since birth, and they're impaired in all kinds of muscles functioning. And there we saw that in patients, placebo versus 119, small cohorts, as you can see in the numbers below, we did see an improvement in it's one of the outcomes. It's how fast can people walk, or how much distance can they do in a 6-minute time point. And there you saw like a big difference between the patients that got 119 versus placebo. And so this gave us some confidence to move, further in the clinical development pathway. But we're still far away. In addition to that, we saw also that in terms of safety, it all looked quite good. So that's another example. And so today's impact. So as I said, we have only one asset, one molecule that is on the market today. But even just with that one, we already have almost 20,000 patients treated. And so that is really, and in diseases where before there was some treatment, but it was not very very well established. And so that is really why we're doing what we do, is to be able to give that impact towards patients. That's what this slide highlights mainly. And our target for 2030 is to reach 50,000 patients, right? So we're also, when we talk to investors or in general, when we say what is our vision, we don't use company revenue numbers to say we're going to reach that amount of $1 million or $1 billion. No, we say we want to reach that amount of patients. That's really the focus that we want to go through, and then the rest will follow. Moving to innovative trial design. So how do we get to patients? It's quite a complex feature, and can be a long way as well. I will give you two examples. And it's the two examples are with trials that led to registration and approval of a drug, of our drug in these indications. And the first one is myasthenia gravis. I briefly highlighted before. It's a neuromuscular disease where you have a lot of facial impacts so ocular, but also in the muscles of the face, there is a lot of problems. There is also problems swallowing, but you also have a lot of fatigue and respiratory issues with this neuromuscular disease. It's also happening at the neuromuscular junction, but in a different way than the previous example I just showed. It's where what happens is that the signaling from the neuron to the muscle is kind of blocked because antibodies, your own body antibodies, are destroying important proteins that are needed for this signaling. And so, for example, the acetylcholine receptor is an important one to make sure the signals from the nerves get to the muscles. Well, if that receptor is destroyed because of autoantibody binding, there is not much signaling happening anymore. And so that's where we thought originally, well, if we use efgartigimod in these patients and all these pathogenic antibodies go down and they cannot destroy anymore that neuromuscular junction and these receptors, then maybe these patients will be better. And so that's exactly what we designed. And now we come to the trial design. So it were in 167 myasthenia gravis patients. They had to have several criteria. Important one is that they had to have, of course, signs of disease, because in myasthenia gravis it's very heterogeneous. You can have very severe type of disease versus almost no symptoms, and so you had to check for that and make sure that you include patients that have an unmet need and can be improved. And then there is, of course, the underlying treatments that they were getting. So usually in these neuromuscular diseases where there is not much therapy available, then corticosteroids are like a typical go-to therapy because it's the only thing that works a little bit. But if you change a lot your corticosteroid treatment dose, it can have a lot of impact and can give confounding results. So they're a very important piece before entering the trial is that there was a stable dose of low-dose corticosteroids, for example. Their patients were then randomized, to either efgartigimod or placebo in a one-on-one fashion. And to get then intravenous efgartigimod or placebo, also in an intravenous way. What is obvious or no, not obvious, that's not the right word. What is interesting in this trial is that it was a personalized dosing approach. That was something quite new at the time as well, is in a trial setting to adapt the dose to the patient, to the individual patient needs. And so that's what happened here. So patients were treated in cycles. So they got the green arrows are like the infusions with the IMP, so the treatment. It's weekly infusions, then you wait a bit, then you do again weekly infusions. So 4 weekly infusions, then you wait again, and like that. So that's where the cycles. And after 3 cycles, that was a maximum, like the minimum amount required, 3 cycles. After that, patients could stop dosing until symptoms came up again. And that was variable for each patient. And then we looked at some typical in MG then outcome measures that we call. It's how to assess that the disease is improving or evolving. And usually in these neuro diseases, they use like questionnaires and scores that assess how patients go through daily living. And so that is actually the MG-ADL, is MG activity of daily living. It's like the key endpoint used and also established by authorities like FDA and EMA to use to assess whether the disease is improving or not. And there, so that was used as a primary endpoint as well. And so what we saw is, of course, yeah, positive results because it led to the approval. But so we did see that, yeah, almost 70% of the patients improved on efgartigimod, and the improvement was, of course, meaningful. It has to be at least 2 points on that scale. And so that was what led to the approval. It was a very clear sign, okay, this drug is making a difference for these patients. And what is also important here is that it worked very fast. So within 2 weeks, actually, we saw these improvements in the most of the patients. That's another picture that shows how the drug is doing its effect on the antibodies and also the clinical scores. So bear with me here, but so there are a lot of colors and lines, but and let me, yeah, they're like this. So the blue colors are the total antibody levels, and the darker color is always the efgartigimod versus the lighter version of the color is placebo. So you see the flat lines are all the placebo pieces, and the ones where you see a reduction in this case in antibody levels. That's what efgartigimod is doing, and that is also translated in orange here in a reduction in the score, which means an improvement in the clinical symptoms. So it's really correlated with the antibodies, and it showed indeed that antibodies first are indeed the culprit here, and that efgartigimod can help. Now, what were the key innovative features of this design back in the time, and still today actually, is that first of all, in line with our culture, the design was co-created with patients and disease experts. So we were again not isolating ourselves and trying to do it all ourselves. No, we went out and asked questions to input from patients and disease experts. This allowed, for example, this adaptive dosing in the cyclic piece. The cyclic piece was based on the second point here, sorry. So that was based on the fact that we saw in a previous study, in a phase 2 version of this study, that actually the reduction of these antibodies after receiving efgartigimod remained for a while, even after treatment. And that allowed to say, well, we don't need to continuously dose necessarily. We can just do in cycles. And that was based on the data that was coming from phase 2. So it was like a data-driven decision to go this way. And yeah, as I said, it allowed, so it's the first time that it allowed for personalized dosing. The impact it also had not only during the trial for patients, because it's really patient-friendly to do so, it allowed also in the approval setting and in the commercialization setting that is also how patients are dosed now in real life because of the example in the trial. And then, yeah, it set the bar for a novel primary endpoint. That's something I didn't mention before, is that it was also the first time that we looked at improvement at a certain way. And it was quite a high bar, and it meant for at least 4 consecutive weeks in the first cycle, the improvement had to be maintained. So that was the primary endpoint compared to before it was you just need to show an improvement on a one-off basis, for example. But since that trial, other trials have used that endpoint as well, that high bar. Another example is in chronic inflammatory demyelinating polyneuropathy, in short, CIDP. It's another neuropathy, neuromuscular disease, actually, where, well, it's very debilitating. Patients that have CIDP, they, I think around 30% end up in a wheelchair. Most of them need a cane or support to walk. Many of the patients cannot use their hands properly and do these daily tasks. So it's very impactful as a disease. It's also an autoimmune disease where we think that, of course, IgG play a role. So these antibodies are attacking the peripheral nerves. So it's not central. It's all in the periphery in the body, where the myelin, that is myelin sheath, is an important structural element in the nerves that leads to fast conduction of signals. If you don't have myelin, the signals would go much slower and would not translate into movements like we are used to. And on top of that, myelin protects the nerve physically from whatever damage could occur. But what these antibodies do, they bind somewhere to the nerve, they attract the whole immune system, and they destroy the nerve in patients with CIDP. So we thought, okay, let's try with efgartigimod. And therefore, we designed the following trial. So that was the ADHERE trial, in which I personally was highly involved in. And so it was a very complex trial. It was also the first time in CIDP that we went from no data at all in that indication. So we had data on efgartigimod in other diseases, but in CIDP, we had no clue, where we went straight to a big trial that would be registrational and be used for on market. So that was also a way to speed up the timeline. And therefore, we took a big risk to organize a big, costly trial without even having any proof of concept before. So what we then did is we built in the proof of concept within the trial. So that's why the design is the way it is. So, well, maybe let's go back here. So the first piece was identifying patients with CIDP. Very important, because in CIDP, the diagnosis is very difficult. CIDP looks like many other diseases that are not CIDP, and a lot of neurologists, they find that a challenge. Sometimes, so I think on average, a patient with CIDP takes around 2 or 3 years before understanding that they have CIDP. So, of course, if you're organizing a trial, you want to make sure you include CIDP patients and not ALS patients, for example. And so that was something that was important, and therefore we included an innovative tool to make sure we have the correct CIDP patients, which I will talk to later. After patients were included and eligible to go into the trial, they passed to a first stage, and that is the open label stage here. It's everyone gets efgartigimod, and it's actually to kind of serves as a proof of concept, where if we see that patients respond to the treatment, only then they were able to go to the next stage, and which was then, of course, the important placebo-controlled phase, where we look for, okay, you had efgartigimod before, but if we now take it away, in half of the patients, do we then see worsening? Because then we can retrospectively say, well, efgartigimod has an impact in this disease. So that's why we call this a kind of an innovative adaptive design, which indeed in the end led to the approval of efgartigimod in CIDP. And then, patients can, after the stage P phase here, they were allowed to continue on efgartigimod if they wanted, in an open label extension stage, which is a quite common thing to do in clinical trials. It allows us to gather much more long-term information, but it also allows for patients that feel the benefit to stay longer on the drug as well. And these were the results, right? So we see that also roughly 2/3 of the patients responded on these endpoints that we also look and that are similar as to the myasthenia gravis. So these are these neurology scores where we test the muscle strength, muscle weaknesses, how patients are moving towards daily life, and those endpoints are validated, and we really see improvements there. That's in the stage A parts. And then when they went to stage B, we did see a big delta, a big difference between patients that were on efgartigimod that were maintaining these benefits versus patients that were switched to placebo and they lost actually the benefit they obtained in stage A. And that's how we then, with high significance, showed that efgartigimod works in CIDP. Now here as well, oh, and I see I have my 1 minute. Over time, what was innovative here is again co-created with patients. Phase 2 design that was straight to registration, that was novel. We set a new bar for improving diagnostic certainty by implementing a committee of experts, worldwide experts, to look at the medical history before patients entered. Now it's being used by all the other trials in CIDP. And it was designed for success by being event-driven, dynamic, and help us to achieve record time completion in this indication. So sorry, I went over time. That's why I'm going a bit faster, but here are the key takeaways. So what is really important at argenx is that we're science-based. We have a unique culture. We stimulate shared ownership. We're horizontal. Humility is really a big piece of it. We empower our colleagues and our employees to create an atmosphere that allows innovation in all corners of the company. And then the partnership with academics, which I highlighted a couple of times, is really a success formula. And then keeping the patients top of mind, which drives how we design trials and how we try to speed up timelines to get therapies to patients. So that's it, actually, from my end.
Thank you, Geoffrey. Thank you very much for highlighting some of the things that I think start really to understand is how important collaboration is with different stakeholders and with always patients at the center of valuable innovation. Definitely, indeed, the academic collaboration is co-creation, as you called it, indeed, very important. And I think what maybe you were too humble is that argenx is really, really a Belgian success story. And I am honestly very proud of it myself as a Belgian person, but also as a European. I would say it is increasingly exciting how you stay as much as possible here in Europe, because indeed I can imagine that the pressure for a biotech company getting funding, which is much more accessible in the US, is very difficult to really stay here. I hope really that the students, well, we will have more modules on market forces and economics later in the next module, but I hope that the students start to understand that indeed the pharmaceutical reform and the European Biotech Act that allow this more adaptive framework and a little less regulatory, while still, of course, keeping safety and efficacy and the basic rules of good clinical trial conduct is key, of course, but that adaptive trials really can make the difference. Not only for rare disease patients, well, I would say adults, but and maybe this is a very nice way to make this transition to our next speaker. As I told, so I will introduce Delphine in 1 minute. But as I told the students this morning, children are not just little adults, and this is very important. It's a very vulnerable patient group, and neglected patient group. Unfortunately, we will hear why from Delphine. I want to, in any case, Geoffrey, you can definitely stay on. I hope. Yes, wait, let me see. I think, the question. Yes, Anna Maria, maybe I can save some time and say, do you think the key takeaways on this last slide is what really distinguishes argenx from other companies and make it currently so successful? Thank you for this question. Maybe there we can let Geoffrey answer and then go to Delphine.
Yeah. Sorry, can you repeat the first part of the question? I missed
Whether
you there.
those takeaways are really what distinguishes Argenix from your competition.
Oh yeah. I think again that's the success formula. For example, on that collaboration piece with academic experts, when we talk to them, when we reach out to experts, they also say, well, it's quite new and they're not used to collaborate with a biotech or a pharma in the way we do. They say that we are much more open and we're more in a partnership type of way as opposed, and that allows, of course, to create many more things instead of having a transactional type of a way of doing. And it's actually very simple, but what we see is that it's not really being adopted by other companies. So I think it sometimes can be very simple. And yeah, true to
I think indeed it's right, but it will take for the bigger pharmaceutical companies. They understand. I think they're not. They see it that it can work, but it takes a lot of time to move,
to change it. Yeah,
to change this whole drug development model that they've been working for and that worked successfully for many, many, many decades. That it's really difficult for them to come to move, to come into motion and to adapt and be adaptive. So I think there indeed you can take the advantage. And with this, I will go to Delphine to give her enough time to present as well. So Delphine is the managing director of one of the founders of the Belgian Public Interest Foundation, KickCancer, the mission of which is to foster fundamental and clinical research in the field of pediatric oncology. KickCancer is achieving this objective by funding research, advocating an innovation-prone regulatory, economic, and cultural environment with the European institutions and the pharmaceutical companies, empowering patients and raising awareness. Delphine is also a member of Childhood Cancer International, Europe's regional committee, and plays an active role in their European affairs, as well as research and innovation pillars. She's the mother of 4 boys, one of whom was diagnosed with cancer at the age of 9, and it was the lack of innovative treatment offered to her son which grown her and her family to found KickCancer. Before KickCancer, she had a career as an international corporate lawyer, strategy consultant, and legal advisor to a private investment fund dedicated to renewable energy. And this is exactly what Delphine will explain to you, that patient-driven innovation is actually, I think, what is needed for to drive indeed or to accelerate the innovation that we as patients we need, but definitely to meet unmet medical needs. Delphine, the floor is yours.
Thank you very much, Hilde, for the very kind introduction. I hope everybody has not his or her head spinning. So my name is Delphine Hielen. I founded KickCancer in 2017, and as you already heard, I did so despite the fact that nothing predicted that I would change the course of my professional career, because I had a son with cancer, and when he relapsed, his prognosis was poor, and still he was only proposed old off-label drugs with no promise of success. And so I wanted to change that, and so I founded KickCancer, and of course I prepared slides for you, so I'm going to start sharing now. And the basic mission of KickCancer is to cure every child with cancer. We want to find new treatments, improve existing ones, and kick children's cancer away, to send it not to the stars, but somewhere in the very dark hole far away from planet Earth. Of course, when I founded KickCancer, I knew that I would be active in a given environment, and so I decided to understand the environment properly. So my slides are not changing, and so here we are. And so this is a crash course on the reality of pediatric cancer in Europe, and I'll give you a few numbers for Belgium as well. The reality of pediatric cancer is that it's 35,000 new diagnoses a year in Europe. Sounds like a lot of patients, but it's only 1% of all cancer cases. So it's 3.5 million new cases of cancer in Europe. And pediatric cancer is not a single entity. It's 16 types of cancer, 60 subtypes. In Belgium, we have 430 new patients a year. And the standard treatment protocols that are ongoing in Belgium today amount to 55. So it really shows you the width of treatments that are needed to treat children with cancer, and it shows you the rarity of cancer. Despite the fact that it is so rare, it is still the first cause of death by disease in Europe and in Belgium for children over 1 year of age, with 60,000 children dying every year. We have reached today 20% cure rate for children with cancer, and it's a stellar improvement from the 50s to the year 2000, when we grew from 50 to 80 to 76%. But since the year 2000, the stagnation of the cure rate is very alarming, and it's actually quite surprising when you know that there has been so much innovation in terms of genomic understanding of cancer, immunotherapy interventions that were developed. And so it's reflected in the number of new drugs that have been approved for children. And these numbers are drawn from a study spanning from 2007 to 2022. And over that period of time, we had 16 new drugs approved for pediatric use, in comparison with 200 for adults in the field of cancer. And so you see that children are quite underserved, and even more so because 8 of those drugs have been put on label for pediatric use, but relate to cancer types that are not extremely relevant for children. And not only do we need to make sure that 100% of the children will cure from cancer, but we also need to ensure that they live a long and happy life thereafter. And today it remains a complicated situation, with 2/3 of the survivors suffering from long-term side effects caused by the treatments or caused by cancer itself. These side effects can span from organ removals, amputations, which are quite straightforward to picture, but also disease that will develop later when the patients reach their 30s, and it can be diabetes, cardiac problems, post-traumatic stress disorders. There are many, and of course, learning impairment, especially in the field of brain tumors. And so we really need to do better also for the patients who do survive. And we see that there's room for improvement, because we know that children who run out of standard treatment options don't often get a chance to participate in an innovative clinical trial. And it's only 1 in 10 children who have that chance. And that's a stark contrast with the adult landscape of cancer as well. And so what does Kid Cancer specifically do about it? We, as a former consultant, I like the matrix of 2 by 2. So we finance research and we defend children, and defending children can take many forms. I'll dive into that. And we're European, and that's extremely important, because when you are in the field of rare diseases, numbers matter. And so size and scale matter. And so we need to work at a European level and join forces. But we're, of course, Belgian as well, and we do everything we can to support the Belgian landscape too. When I took the decision to found Kid Cancer, maybe some of you have been in contact with the field of philanthropy, but anyone with a good idea can start his or her new venture and collect funds and ask for donations from people's generosity. But it's not always extremely efficient to add players in a crowded field like philanthropy. And so I really wanted to make sure that there was no actor that ticked all the boxes that I wanted to see filled when we founded Kid Cancer. And so we wanted an organization that would be independent. And by independent, we meant independent from industry. Patients' organizations receive a lot of funding from the industry. It's definitely something that we wanted to be freed from, but also independent from research institutions. And a lot of parents or survivors' endeavors in the field of research are very closely intertwined with the hospital where the patient was treated. And I completely understand the emotions and gratitude, which I do feel towards my son's doctor. But the independence towards institutions is also the key to making sure that we do not duplicate research, that we select the best research projects, that we also move the fields more broadly than just supporting one single institution. Although we are not specifically connected to one hospital, we collaborate with everyone. We collaborate with the industry. We don't take their money, but we speak with them on a regular basis. We talk to all the other NGOs and we try to converge wherever we can and stand behind policies that we are aligned on. And we talk, of course, to every research institutions and all the hospitals with which we have topics to discuss. We are not limiting ourselves to funding research. We think that if it was just a question of money to solve and cure children with cancer, it would have happened a long time ago. Nobody likes children suffering. Nobody wants children to die from cancer. We know that if it's not sorted out yet, we know that it's because the world is very complex. And so we're looking for structural solutions. And so not only do we finance research, but we also plead for better laws. And you will see that we also try to change how research is conducted. As I already said, we fund research in Europe, not only in Belgium, but we also advocate better laws in Europe, not only in Belgium. And I will give you more information on that in a few minutes. And so to sum up what we do research funding, we advocate a better legal and cultural framework, we empower patients, and we raise awareness. I will not talk about awareness because it's not relevant for you. But so we drive innovation by funding research like activists. In every single thing that we do, we try to change the way things are done to make them more efficient. And so when we started funding research, of course, we didn't have our own selection process, but it was the first project I gave myself when I founded Kick Cancer. And we started with 3 NGOs from France and Luxembourg, but now we have a group of 5 funders. So we added the Netherlands and Spain, and we created an international non-for-profit organization together called Fight Kids Cancer. And we fund research together, and you will see that it has an impact, positive impact on scale. Our international NGO is designed to welcome new members because we think that, of course, we can all start fundraising more and increase our impact by being better fundraisers, but it's even easier if we convince others to join us so that we can catalyze funding for pediatric cancer research. We have given ourselves the goal to accelerate research and innovation, and we have done that by organizing annual recurring calls across European borders. And it sounds very straightforward and stupid, but before Fight Kids Cancer, researchers who wanted to collaborate, which they had been doing forever across countries, had to look for funding in each of their country. So imagine a project with 5 research teams. It means 5 calls for funding. They have to submit their project to 5 grant contracting, 5 progress reports. Imagine that one of them doesn't receive the funding, then either the project is jeopardized and cannot go through because one of the key expertise is missing to the project. We fund 5 countries all at once with 1 contract, one process of selection, and 1 project report on an annual basis. So we offer a one-stop shop. But more than that, we come back every year. And we are tailored to the needs and the understanding of the pediatric cancer landscape. The research teams also had to submit their projects either for specific once in the lifetime call dedicated to pediatric oncology, and they had to bundle up a project very quickly, or they were submitting their projects in general cancer calls, and the understanding of the needs of children with cancer was poor and their chances of success were low. So basically we see, thanks to the fact that Fight Kids Cancer has been on for 6 years now, that the quality of the project is improving significantly over time. We see more new ideas being submitted, and so we see a tangible impact to what we've been doing. Fight Kids Cancer is also funded by patients' organizations for patients, and so we include our political wish lists like data sharing, open source publications, but we also value impact a lot, and we make sure that impact is not disregarded versus exciting science. If we need to create a database to pave the way to new findings in certain disease areas where we know that the understanding is so poor that we have to start with a database, we are willing to fund something that nobody finds sexy. We are by patients for patients, but we have a venture with the European Science Foundation, and they run our selection process with independent international experts. They played a super important role in ensuring that we would avoid the conflicts of interest that were congenital in a field of rare diseases where everybody knew each other and people thought that it was normal to be an evaluator for a project of a former colleague or a good friend. The European Science Foundation keeps the church in the middle of the village, as people say in French, and makes sure that we have a conflict of interest-free selection process. And that has also changed the practice. So without even looking yet at the scientific foundings that we have delivered with Fight Kids Cancer, we have changed the landscape. And numbers also show this change of landscape. As you will see, we started with 3 partners, and we were fundraising 3 million, but that was COVID years. And we could barely increase in the second COVID years. But once we started adding more partners, we jumped to 7 million. And then unfortunately, we had an output problem. We had too much money for the projects that we received. And so we could not we couldn't invest all of our money in 2023. And so we had to think about what can we do in case of output problem? How can we invest more in research? And then we could continue to grow nicely in 24 and 25, and we're set probably for about 15 million for 26. So we will have crossed the mark of 50 million euros over 7 years, which I think personally is quite exciting. A few things that I would like to discuss with you is we realized after 3 years, we made a first post-mortem of how have we been doing in terms of project selection. And we saw that we have a disease-focused problem. And then, of course, in 23, we had an output problem where we could not invest our money. And so I'm going to show you how we reacted to that and changed the way we decided to fund research. So first of all, we realized that we had to make pediatric cancer sexy. So make peds attractive again. So it's like MPA. It doesn't sound as good as some other political movements, but I knew it sounded bad when I made the slide, but I still wanted to keep it that way. So we know that pediatric science is structurally less attractive than adult science. Children are healthy, it's a good thing, but when they're not healthy, it's really a double loser situation. Adult oncology structurally attracts a lot more funding than pediatric oncology because there is a financial and commercial market to develop drugs. And so if you want to do research in oncology, you will be naturally drawn towards breast cancer, colon cancer, lung cancer, and so on, because it's profitable. It's also more difficult to conduct research in areas of rare diseases because attracting or collecting biological material is difficult because there are few patients and samples are scattered across many countries. And so the research community is very small in pediatric oncology. And if it's complicated for teams to get funding for projects, it makes it also more difficult to hire PhDs and to keep postdocs. And so we did see that the sort of feeding pipeline for research was a bit complicated. And so we decided to be intentional in our desire to attract researchers. And we started in 2023 a new funding opportunity where we would promise researchers the ability to build a lab and hire long researchers, and so create long-term career prospects. And so we also thought that maybe we could fund things like tumor libraries, structural needs. And so these were the two options that we looked at, and we went for the first one, the possibility to create a pipeline of talent. And so we created the Innovation Award. And we started that together with an American foundation. We fund it, but they run the selection process, and they do so because they have the expertise in that. They have had an Innovation Award in the States for many years, and so they know how to do it. And it's a €1 million funding over 4 years. And it's not earmarked money. So basically, we give a talented researcher based on his or her track record, not based on what they will want to do. We give them 1 million over 4 years to fund their scientific dreams. And it's a very light selection process. Basically, you can complete your application form within 24 hours. You need someone to sponsor you, a senior person, and then if you are selected for the second round, you just write 3 or 4 pages with your scientific dreams without being too technical into how you want to do it. Basically, we have already awarded 2 innovation awards, and we pay a PhD student, we pay the salary of a postdoc, we buy computers, we pay conference travel costs. We really pay whatever comes along the way in terms of need of a lab. So it's really very empowering for someone who wants to start a team, because it gives them the bandwidth to apply for funding for projects. Our funds are not earmarked, they do whatever they want, but then they can get more funding for more projects, and it really creates a virtual circle. We also decided to be critical on how we were doing in terms of serving the patients that we want to cure. And so after 3 years, and you don't need to look into the details of this, but we looked at how well we've been doing by disease to select research projects. And so in 3 years, we had selected 25 projects. We have a 2-step selection process. We receive 133 applications. We selected 75 to move to the second phase that had to fill out a full application. And so we ended up with 21. And so what we saw, if you look at the color bulbs, you see that we had received a lot of applications from brain tumors, but we ended up funding very few of them. We had received a significant number of neuroblastoma applications, but in comparison, we had funded a lot of them. And so looking back critically at, it's neuroblastoma that was so intensive, 21, and it's not a very common type of pediatric cancer. So basically, we had been overly serving the community of neuroblastoma, basically because they are quite advanced in scientific terms, and so they have more exciting research projects. But the brain tumors and the sarcoma patients were completely underserved, despite the fact that they are the 2 deadliest types of pediatric cancer with the most severe long-term side effects. So we really thought we're not doing our job well. And so we decided that we would organize brain tumors only and sarcoma only calls in 23 and 25. So the brain tumor score process is completed, and we see that the seeding effect of organizing this was brilliant, because we receive now more application from brain tumors. So we see that it had a positive impact on the community of the brain tumors scientists. Of course, we went back to a general call because we don't want to forget the other patients. And now in 25, the sarcoma call only was launched, and we will do the performance selection in 2026. The reason why we went for brain first, if you look back, we had a lot of application in brain. We saw that the research community was very active and able to deliver for brain tumors only call quickly, while the sarcoma community was a lot less active and needed to have some encouragement to submit more applications. And so we used the time between the moment where we decided to launch this call, so between 22 and 23, we listened to the needs of the brain tumors, and we sort of redefined the selection criteria and the specs of the projects based on the needs of the brain tumors. And we used the 3 years to actively meet with the sarcoma community and also ensure that pediatric researchers start collaborating with adult researchers. And we have received, that's actually quite impressive, an avalanche of projects in the sarcoma core. So we hope that it will also have a long-lasting effect. And so now switching to advocacy, how do we do that? Because we are also very prone to influence the regulatory environment. We do that at a European level, and we do that on the Belgian level. In Europe, we work, of course, with a lot of partners. First of all, Key Cancer, my Belgian organization, is quite central in the advocacy work at the European level. We are based in Brussels, close to the European Parliament. I have a background as a lawyer. And so it gave me a natural instinct to move into that direction. But of course, if I want to represent pediatric cancer at a European level, I need to be legitimate and represent the voice of children from across Europe with very different realities, Eastern Europe being very different from Belgium, small countries, big countries. Basically, Sweden, it's such a huge territory. They have a very different reality. We have to reflect that. So we work with the European Patients Umbrella Organization, and they have 2 lobbyists in the Key Cancer offices, and I work hand in hand with them. And actually, I oversee their work. We also work with doctors very closely. So the European SIOP Europe, which is the International Society for Pediatric Hemato-Oncology Europe. And we work with other patients' organizations, Eurordis, which is the rare disease organizations in Europe, the Anti-Cancer Fund. We work with WeCan, which is a patients' organization for cancer, but all cancer, not just pediatric cancer at the European level. We have worked on many regulations so far. We worked on the clinical trial regulation, and we were really excited to see some simplifications. A bit less excited when we saw the actual implementation of those simplifications. It didn't seem that much more simple than we were hoping for. But we're working on a new wave of amendments to the clinical trial regulation in the framework of our work on the Biotech Act. We also worked a lot on the pharmaceutical package, and we worked specifically on 2 key changes for pediatric cancer. The first one is what we call the mechanism of action. Adult drugs must be translated into pediatrics by principle since 2007. But that basic change, which delivered wonders in cardiac disease, inflammatory diseases, rheumatology, or diabetes, actually delivered nothing in cancer. And why is that? It's because the obligation has a few exemptions, and one of which is that if the disease does not exist in children, you don't have to investigate it in pediatrics. Totally makes sense that we don't start developing drugs for dementia in pediatric in general. Unfortunately for cancer, even if children don't get breast cancer, they do get sometimes cancer types that can have a mechanism of action similar to the mechanism of action of the drug that is developed for breast cancer. And so children were missing out on many drugs that were promising for them. And in the pharmaceutical package now, the exemption is limited, and the European Medicines Agency will have the opportunity to mandate the market authorization holder to investigate pediatric cancers if the mechanism of action is relevant for a pediatric cancer type. And so that's a huge win for us, because it will prevent a lot of off-label use. It will also speed up access to new molecules, and so we're extremely happy about that. Another dimension that is a great win for rare cancers like pediatric cancer is that there is now a specific line for academic drug development. And by that, we know that in pediatrics, in pediatric cancer, where the mutational burden of the tumors is quite low, monotherapy never works, and we need combination trials. And we know that combination trials are very challenging for the industry if the drugs that you must combine are not proprietary for them. In that case, academia can offer a quick and easy solution by running the trial themselves with the drugs that are sometimes off patent or chemotherapy combined with a new intervention. The problem is when you have an academic trial, the drug ends up being off-label, because only the market authorization holder can apply for a label extension with a European Medicines Agency. And if you have an off-label drug, in many countries you end up with an access problem, because social security systems do not reimburse off-label drugs. And so now we have obtained a specific track for academic drug development. If academia sets up a trial and collects data in a fit for filing fashion, they can submit their data to the European Medicines Agency. They review the data, and they can then mandate the label extension to the industry, to the market authorization holder, which means that the market authorization holder doesn't need to run any additional clinical trial. They only, I know it's a big only, but they only need to fill out the steps for the label extension, which is a purely regulatory process. And I'm quite confident that with AI today, that can be quickly fixed. And so that was a major change. By the same token, we do work with academia because we know that they're not very good in their data collection process. And so we set up working groups, which we called Fit for Filing, to train them, encourage them to collect the data in a way that will make it possible to have them approved by EMA. So basically, we worked at the regulatory level, but also at the cultural level with our researchers. I mean, that battle isn't won yet, so I'm not going to be too triumphal right now, but we're working on that too. We also advocated quite strongly a change in the health technology assessment, and now pediatric cancer and rare cancer are pioneers, and they are assessed for their future reimbursement at a harmonized European level, which makes it possible to work in parallel instead of sequence with the EMA. And so hopefully when a drug is approved by EMA, a good deal of everything, all the requirements for the health technology and future reimbursement of the drug will have been completed simultaneously. And we not only did we change the regulation, but we were very active in scouting for the patients that participate in the assessment process. And so we are active on both fields there. We're working actively on the Biotech Act. We're also working on the regulation called the European Research Area. extremely important in rare diseases, that diplomas, mobility, incentives are aligned. We're trying to push innovative funding models. Everybody has heard about the public-private partnerships between industry and European funds. We're trying to work on philanthropy, public funding, because we know that the industry is not very likely to come to fields like pediatric cancer, so we're active on that as well. And we're working quite hard as well on cross-border care in rare diseases. Patients need to travel to access care, but sometimes also clinical trials. It's a whole conundrum today to have the S2 form, which is what you need to have your participation in or your treatment reimbursed when you travel abroad. We're working on that as well to make sure that the cross-border care directive includes participation in research as well. And in Belgium, we also work with other organizations. Kinderkankerfonds, Tukkie Sunchild, and Aide aux Enfants Cancéreux are the organizations with which we work the most. We also work on a day-to-day basis with Foundation Against Cancer and Kom op tegen kanker. And we work naturally with the Belgian Society for Pediatric Hemato-Oncology. So you find back the patients' organization first, which are quite small in scale, but they have very good knowledge of the field. We work with big players on many advocacy topics, and we work with the doctors. And so what have we achieved? We twisted the arm of the Belgian government to make sure that Ukrainian refugees could be evacuated to Belgium and cared for here. We were shamefully the last country who hadn't organized a trip to Belgium for patients. And so it was a real activist negotiation. We worked towards the reimbursement of off-label drugs in pediatric oncology. The field is, I'm going to come back to that one later. I'm going to show you how we worked to get this done. So I won't expand on that now. We work on the organization of care for adolescents and young adults. Again, I have specific slides on that. And we work on, we obtained the increase of the requirements for specialized pediatric oncology units. Now they have to have 100 patients per year, new patients per year instead of 50. And that is, of course, meant to increase the quality of care because nothing replaces expertise. We also work at the very Belgian level on the topic of the access of international trials, and we are involved in the drafting of the new Belgian cancer plan, both for children and adolescents and young adults. And so what did we do for off-label drugs? Basically, the field is 50% of drugs in oncology are off-label, 50% of drugs in pediatrics are off-label, and nobody knows how many of them are off-label in pediatric oncology. But the impact is very well known. Many drugs are not reimbursed, and the impact for healthcare professionals is huge because they need to find alternative routes for the reimbursement, and it's a lot of paperwork. And so we built a case for the problem. So with a consultant, we put numbers on our reality. So what is the actual cost of the drugs? What is the actual number of hours lost by healthcare professionals? And then we made a whole projection for the country based on a partial database for the cost of drugs. So we had the data for Ghent. They had been very thorough and punctual in collecting how many off-label drugs had to be paid by the patients. And we worked on the database of the cancer registry, but it was a huge, complex work because the dose, they are per square meters, by volume of patients, but we know that a 2-year-old does not receive the same amount as a 12-year-old. So we had to take into account the spread of the disease. And so we had a pro bono consultant making us a very nice database. And then we worked and conducted a survey with the doctors, social workers to quantify how many working hours they were losing trying to look for funding for drugs which are standard of care. And so these are all drugs which are noted as being the best known treatment for a given indication. We have European protocols for pediatric cancers. And so we really wanted to show these people are not working to make wonders. They're working just to make the machinery work as it should. And so with the pharmacists, we identified a logic under the problem. We saw that not only were there off-label drugs that were not reimbursed, but also drugs that were on-label but not reimbursed in Belgium. There were drugs that were not authorized in Belgium, but they were authorized in Europe, and we had to purchase them in our neighboring countries. Some of these drugs are standard of care. Some of them were maybe not so standard of care, so we took them out of the list. Are they treatment based or are they support based? And so we ended up with a list of drugs to be reimbursed and a framework to classify them. And we used striking examples to illustrate our plea. We have Julia, she's 17 and a half years old. She received irinotecan for 12 months. First 6 months, it's not reimbursed. She turns 18, it's reimbursed. Basically, that made it very clear that there was no logic in not reimbursing the drug for a given treatment. And so we framed a future-proof solution because, of course, we could have like a bulk of 50 drugs authorized or reimbursed at once, but standards of care keep evolving. We have new drugs off-label, not reimbursed, adding to the standard of care. We wanted to make sure that we didn't have to redo this whole work every time. And so not only did we have the backlog of 50 drugs approved, but we have a pragmatic, future-proof solution with annual meetings with the Interdisciplinary, and we discuss all the new drugs that have to be added to the list, and it works quite well. So we had an article published in a scientific journal prior to the discussion with the Interdisciplinary and the cabinet, but we also had a press campaign to make it very striking, and so it's all sorted. For clinical trials abroad, we also work currently with the cabinet, and we know that there is no commercial interest, as I already said, in pediatric oncology. And so there is a lack of development. Pediatric cancers are very rare, and clinical trial, especially early phase clinical trial, recruit very few patients per year in Europe, 15 to 20 per year maximum. And so the trials are run by academia, and so they're funded by philanthropy and usually repurposed drugs. So the budgets are small, and it's necessary to centralize the trials for cost purposes, but also to make sure that we centralize expertise. And so that boils down to children from Belgium hardly ever get a chance to access clinical trial in Belgium. And so here we use a completely different approach to build our case. The S2 form regulation to travel abroad, it's a nightmare to understand. These are 200 pages regulations with guidelines to issue the forms. And so we spoke with a specialist. And so Solidaris was very nice and actually had several meetings with us. We described our working cases to them. They helped us understand the subtleties of the regulations, and they helped us analyze solutions by problem type. And so we co-created pragmatic solutions for each case to help fragile patients. And so then we were able to work with patients to show the dangers of the statu quo. The actual danger is, maybe you heard about those, but there were fundraising and media campaigns for patients who had to travel to Rome in the course of 2025. And these are patients who needed 330,000 euros to participate in a trial. So you can't just find it with small fundraisers. And so with the sort of public interest problematic and the support of a health insurance company, to build a very strong case to propose pragmatic solutions, we had a chance to meet with the Minister of Health, and they are now reviewing the guidelines, and hopefully in the course of 26 it will be past history for patients. We also work on the organization of care with IAS, but I'm a bit short on time, so maybe I will switch this case. You will have it here. I'm sorry. I wanted to show you another example of what we do, because I think it's completely different. It's not about politics, and it's what we do with the research fields and how we try to influence them. Basically, we, as patients organizations, started to have really problems about how research is organized or research is funded. And we did see that it tends to work by, oh, we have scientific leads, and so we go towards the scientific projects. While, of course, we would like a patient-centric strategy where we go from unmet medical needs and then we head towards research projects. And that's how we would like this to work. And so together with other patients organizations that fund research, we made a plea in research groups to say, we want to support research projects where they are needed the most. And so we really showed the framework we would dream of is clarity on actual event-free survival, overall survival by disease group, a clear understanding of what is the current treatment, how does it work, is there a curative standard of care or not, understand where we are today, how much do we publish already on the disease, is it a completely forgotten disease, do we understand the biology, do we have biomarkers, do we have leads for clinical trials, do we have alternative drugs to the current standard of care? Are there standard salvage regimens? What are the challenges? Is it blood-brain barrier for brain, cold tumors for sarcomas? And then define what do we need to improve for each disease? Do we want to improve overall survival because we're nowhere? Do we want to go for event-free survival? Is it the long-term toxicity that is terrible? Is it the acute toxicity? So basically, we wanted to create a framework where we would understand for each disease exactly where we were. And it's quite striking to see that very often scientists and clinicians would tell us, yeah, but it's clear, it's in our head. But then when we said, write it down for us, it was a lot more complicated. Basically, we wanted to end up also, you know, a 2x2 matrix. Sorry about that, but a matrix where we would have low medical needs, the understanding of the disease. And so, of course, here it's slow, but we understand the disease well. Here we understand the disease well, but it's high unmet medical need. And so we think that we can move fast, but here we see high unmet medical needs and we don't understand the disease very well. So here we need to go for fundamental research. And then it went from wish to reality. There is a concept called Cancer Grand Challenge, which is a transatlantic funding opportunity in research. And it's a 25 million research project. It's about drug development in pediatrics, but there are always patients' work packages in these projects. Of course, the biggest share of the 25 million pounds is going to research, but there is also one work package on the prioritization of the diseases. And so we run so we didn't do it as thoroughly as we would have hoped, but it was at least a quick win and a very pragmatic way to move forward. We organized surveys with doctors. We identified 13 diseases with the highest unmet medical needs, and we are organizing workshops to identify what do we understand exactly today, which drugs should be developed. We know the targets, but we don't have the drugs yet because we don't know how to develop drugs for this type of targets. How do we see a plan for that? And then also identify drugs that already exist for which we can already start setting up clinical trials if that's the case. The last work package is a business model for drug development in non-commercial diseases. And so it's basically a work package where we have funding to think about how can we improve efficiency in drug development to lower the cost, how can we accelerate development pace to help patients faster and reduce the cost because time is money. and what is a realistic drug development budget to reach affordable, efficient cures, and then how do we have to align philanthropy and everyone to make sure that we move forward. And so that's the slide of the workshop I participated in on rhabdomyosarcoma. And these are, so the introduction always comes with a patient, and it's the last thing I will talk to you about today. But we explained the researcher, that's your perspective. You have a piece of tumor here, but that's our perspective. Sorry, there's a picture of my son. Of course, for me, it's always a bit hard to think about it, but they have a real life. And basically, the ultimate goal is to keep these people alive. And then we also showed them that's basically, you know, what Christopher received in terms of treatment in '86. And if you look at Alice, who started her treatment in 2015, we see that basically VAC is almost the same treatment regimen as what Alice had. Well, she didn't have organ removal, which was extremely brutal and left Christopher with a terrible quality of life. But we see that the treatments that patients receive over time do not evolve at the same pace as other technologies, and that's what we wanted to underline. We put pictures and quotes of patients to make it tangible for research, and we also have clear asks. We say we want faster cures, we want a plan for funding organizations, and we're intentional on how we want to reach what we want to do. And so I had slides about patient empowered and showing you the tools that we have, but you'll have to read it. I'm really sorry about that. The only thing I want to tell you about is it sounds like we're doing so many things and it's so successful, or maybe it doesn't sound that successful and I'm a bit hypomaniac, but I want to show some of our struggles as well. First of all, it's extremely difficult to fundraise for advocacy. Companies don't want to fund lobby, it's ugly. They want tangible projects, sorry for the typo. So they want us to come with a book, they want us to come with a trip or a workshop, but they don't want to fund advocacy. Then there is a problem of time and desires. We need to work with patients to do the advocacy work that we do. Patients have already through a lot. Some of them want to give back out of thankfulness, but they tire over time. And so keeping people expert is complicated. And also it's very complicated because they often have another day job. And so they don't always want to give it up. And you will see that the legal framework doesn't make it easy for them to give up a day job because we don't have enough work to hire a professional patient. And so we have a legal framework problem because paying patients who have another day job is challenging. Hiring is difficult. We don't have enough work for one person full time. The volunteering status is limiting. Putting people to work for free is not a valid, respectful option either. And so we're really struggling to engage with patients and make sure that we keep on making their voices heard. And so with that, I wanted to thank you for your attention. And if you have questions, I'm more than happy to take them.
Thank you, Delphine. Thank you so much. If there are questions, don't hesitate to put them in the Q&A or raise your hand and put on your camera, because without putting on your camera, I cannot see you that you raise your hands. It's for me. Did I hear something? No. For me, it's amazing, Delphine. I know you already for quite some time, and through you, I got to know very interesting, very, very bright people, like some dams, amazing. And what I told the students, or where I ended the students 2 weeks ago on Friday, is for me what you every time show again, that passion can drive people, and that you create projects for me that have a true impact on patients, which I think is amazing. What the first question when you explained the KIC answer, your organization was, what can I ask if there are no questions of the student? Do you already have tangible results? And then you just came with slide after slide. How you changed the field it's incredible, and I cannot stress enough to our program participants that indeed passion is what can, yeah, the sky is the limit if you are driven by passion. And I think this is clearly what you showed, interdisciplinary collaboration. I think indeed you said it, from your passion as a mom, but through your knowledge as a lawyer, because I wanted to say also to the students, the talk or the presentation from Geoffrey Istas from Argenic might have scared the economists or the managers in the audience. It's not really about the technical insights. I think you understood by now that what we want to show you is that whatever your background is, you can have a role. I also want to say another thing. The time that we go over time with speakers is time that I will recoup and give back to the program participants. So we will end a little earlier this afternoon. This is my way of making up to them. We have a very short lunch break just because it's a Friday and we want to give you at least an earlier weekend. Delphine, it's amazing. What can I ask you is, because in the Biotech Act, and we already discussed this morning with some speakers, there is a lot about regulatory sandboxing, and I'm really hopeful there, because this gives a little bit of playground. I would say it's still controlled, as I explained to the students, but to move away from this incredible strict regulation where we need to act for safety reasons. But this is something that we could use, this regulatory sandboxing, to give adaptive clinical trials to children too, or?
Yeah, that's definitely the direction in which we would like to go. We definitely hope to have more lenient clinical trial designs to be able to use real world data. Pediatric oncology being so rare, 90% of the children in Western European countries are treated in the framework of a clinical trial. So it's not even completely real world data. It is actually data collected in the framework of a clinical trial. So we do hope that we will be able to structurally use historical control data instead of having randomized trial, especially in those disease areas which have a very poor prognosis, where not giving the chance to the patient of access of the new compound is a bit grueling. The other things that we hope, there are also provisions on administrative support and coordination at either coordination at the national level for European partners, but also sometimes coordination through a European partner. So we do hope to push that for rare pediatric diseases, because it wouldn't make sense to limit ourselves to pediatric cancer, but we do hope that for rare pediatric diseases, we would receive access to this administrative coordination as a standard, because indeed academia or researchers from academia cannot be expected to become as versed into the regulatory science as you would be if you work in a company. And so support is definitely something that they need very structurally. We also want to push, because we are in a situation of market failure, so we want to push provision agreements. And so we want to secure access to the market and basically obtain commitments from states to purchase the drugs before they are fully authorized, because then it really improves the business model of the drug development. If you already know the price tag you can put, so it's procurement contracts, so what I was looking for. So we are pushing the concept of procurement contracts for the new Biotech Act as well, because we think that would be a game changer.
Absolutely. I think this is what companies are searching for, is a little bit more certainty. And I think they're whatever Trump does play at least for once in our advantage, that if we could push giving them a little bit more certainty by allowing a little bit uncertainty and this real world experience too, and this early access to drugs and providing them indeed with the guarantee that we will create a market. I think this is the way to go. Definitely for rare diseases. Well, rare pediatric diseases or pediatric cancer is by definition rare disease, but definitely for ultra rare diseases. I thank you so much for all the work you do. I'm as you know, I already told you, I'm very, very honored that you are here addressing the students, hopefully inspiring them to indeed also step up and help fighting for definitely the most neglected patients. Thank you so much, Delphine.
And good end of lessons for today. Bye-bye.
Thank you. Bye, Delphine. And for all the rest, we will come back at 1 together. And I can say that I hope, well, I will check myself, that the problem on the Université Virtuelle with the slides, accessing the slide, should be fixed. So I hope that this is fixed, and we will come back at 1. Enjoy your lunch. Bye.